Hello, I'm Dr. Tim Jansen. I'm a family practice doctor here in Portland, Oregon, and I've got an intense interest in genetic genealogy. So I've helped organize and lead the advanced genetic genealogy classes for the Oregon chapter of the International Society of Genetic Genealogists here in Portland. These classes are typically held at the Genealogical Forum of Oregon, but secondary to the COVID epidemic, our classes have recently been held online via Zoom. I want to introduce three excellent presentations about mitochondrial DNA that were given for our meeting on April 20, 2021. The first two presentations, given by Dr. Miguel Vilar and by Dr. Paul Maier, provide important background about mitochondrial DNA and its role in population genetics. The third presentation, given by Roberta Estes, focuses on case studies where mitochondrial DNA was used to help solve genealogical problems. Roberta also provided a brief summary of the Million Mito Project at the end of her presentation. If you would like to download the PowerPoint slides of these presentations for your personal study, I've provided those for you here. You can download them and review them at your leisure. Some additional background about Miguel Vilar is included here for your reference. Additional background about Paul Maier and his work at Family Tree DNA and population genetics is also provided here. Background about Roberta Estes, including her blog at DNA Explained, is provided here. With this background, I would like to go ahead and simply move directly to the videos of our presenters, and that will follow subsequently in the next section of this video.
Well, thank you very much for having us this morning. My name is Miguel Vilar. I'm a genetic anthropologist, and I'm part of this team right here that you see listed on my opening slide. Dr. Paul Maier, you'll be hearing from shortly. Roberta Estes as well. You'll be hearing from her and how to use mitochondrial DNA and genetic genealogy. And the fourth in our group is Johan, who may come in as well and say a few things. And we're a group that collaborates, specifically been collaborating around mitochondrial DNA for about a year now. And we're here to talk specifically about that amazing molecule. So what is mitochondrial DNA? Well, I can tell you a few things about it. You'll hear from it for the next 3 hours, but it's an amazing part of your DNA, very small, composed of just a handful of genes, only 16,000 points of data, 16,569 to be exact, inherited strictly matrilineal. We used to say maternal is matrilineal, down the mother's line. What that means is that all of us carry it, 100% of humans, 100% of animals, and even plants have mitochondrial DNA. But in the case of humans, and in most animals works this way, we inherit it only from our mothers. And our mothers, got it from her mother or their mother. Therefore, you can use it to trace inheritance through a single line of ancestry, a direct line of ancestry without any mixing. Why does it only come to the mother's side? Because as you see my little image there, a sperm fertilizing an egg. Well, the mitochondria that actually gets passed on is the one in the egg, not the sperm. There have been some data showing that mitochondria in the sperm can be passed in. There is mitochondria there, but that's rare. It is the mitochondria in the egg that makes up the mitochondria, the zygote, embryo, fetus, and eventually person who that becomes. Okay, so that's why it's passed on. Only through the mother. Daughters have it, and daughters will pass it on to their children. Sons, like myself, also have it, but we're dead ends for mitochondrial DNA. Hence, it is a story of womankind, of our ancestors through a maternal or matrilineal line of ancestry. And it's an amazing story. And if you think of it that way and use it with those caveats, it can tell you so much about the history, about the past, about genealogy as well. So just like I said, it comes from only one parent, therefore it's known as uniparental data or uniparental genome. It's part of that, Y chromosome being the other. A lot of you are familiar with the Y chromosome. That comes strictly from the fathers, but only half of us carry it. My wife doesn't have it. My daughter doesn't have it. My mom and my sister never had it. So half of the people in the world don't even have a Y chromosome. Very informative the same way, but the mitochondria, I would argue, is even more informative, easier to study, plentiful, and we need to give it a lot more credit and a lot more research focus to go into that direction. As you know, the four of us here are huge fans of mitochondrial DNA. So here's a chart again, how it's inherited. I'm the son at the bottom, and I have the blue Y chromosome I got from my father, grandfather, great-great-grandfather, and that little red circle, that's the mitochondria that my sister and I both have. We inherited from our mother and our grandmother and our great-great-grandmother. And although my 8 great-grandparents all have mitochondria of different colors, a rainbow of mitochondria, who are a product of all the 8 of them mixed together, my mitochondria is not mixed. It came from just that one, the mother's 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 mother. Got it? And so it doesn't mix. That's the beauty of it. It's passed on from mother to daughter and from to son as well, and then from daughter to their children. Strictly matrilineal. Very common, very plentiful, hundreds of mitochondrial DNA, each mitochondria, and thousands of mitochondria in a cell. It's so plentiful, short, sturdy, very great to study in the lab. And we've known about it now going on 40 years. So it's incredibly informative. And actually, the way it mutates and the way the mutations are passed on and shared is very interesting and very quite straightforward to follow. We do that through what we call mitochondrial. So again, mitochondria has many mutations. Part of it actually is we call the hypervariable region. It has mutations that go faster. We've been studying those for 40 plus years. Very small, 16,500 bases, and very numerous. I mentioned all this already. And this is what a mutation would look like. And the original sequence had those few letters in a row, T, A, A, C, T, G, C. Somewhere in the DNA is passed on in that egg cell. The mutation occurs to turn a T into a C. That's called a transition, a quite common type of mutation, TAACCGC. Therefore, that 1 point of change gets passed on from the mother to the daughter. Obviously, it's happening in one cell, but it's passed on. It happens in the egg cell to the next generation and so forth and so on. And we look for these mutations, these accumulated mutations that have been happening since the dawn of time, to categorize people into groups. And the groups, we call them mitochondrial DNA haplogroups. So mitochondrial DNA haplogroups really are your clan, your group of relatives, but relatives for one condition, one caveat. They're your maternal or matrilineal relatives. Those are the ones that belong to your mitochondrial DNA. So when I first studied this back in the early 2000s, we were focusing on an area called the hypervariable region. It's about 1,000 or 1,100 base pairs, so about 1/16 really of the whole mitochondrial genome. But it included many mutations, and a lot of them sturdy enough they actually define different branches, and those branches are the ones we call haplogroups. So lineages of shared mutations, especially those consistently sturdy ones in the hypervariable and elsewhere as well, are sort of the key ones that define the branching throughout the history of humans, and therefore the history of the various different haplogroups of humans. Let's focus on A, B, C, D. These are the first of the alphabet. Actually, coincidentally, were the first to be named in the early 90s, and also are the ones associated with Native Americans. So you have mutations, say 16,223, 16,290, 16,319, and so forth and so on. You will be group A. However, within group A, you have to have all those plus a few more, 16,111 and 16,362, to put you in the next level, A2. So B works the same way, C and D. You look for the mutations, you look for the shared mutations, and then your subgroups will have the same ones plus more. So the letters of the alphabet correspond to known mutations that have been known assigned to have occurred and to occur more frequency, higher percentage in parts of the world. Like the groups A, B, C, D, defined by those mutations, are most common among Native American groups. H through K, N, T through X, those are very common in European, Western Eurasian groups. L0 through L, actually all the way up to L7 possibly, or L5, 6, 7, occur in African groups, and lots of the other letters are more common in Asian and Australian groups. And you may have seen a map like that. We have similar maps for Y chromosome that shows both the phylogenetic tree, how the groups are related, how they came, how they evolved, when and where, and also where they're found today. So you'll see maps like that probably throughout this talk. Again, when I was doing this early on, we were looking sometimes just at the haplogroup level, trying to find specific mutations so you could place a DNA sequence in a haplogroup. Nowadays, we actually will find those and we'll try to find the next level branch. So if you've taken a mitochondrial DNA test, you may know, like I showed you in our first slide, you may know a lot more than just I'm haplogroup J. You now know I'm haplogroup J to C 1B, and so forth. It goes letters and numbers, sometimes going 5, 10 levels down. And this is how we do it. We categorize them at different levels, at different breaking points in the evolutionary tree. What we've done beyond that, we've been able to categorize them geographically as well. And this is sort of a summary of where you find various different haplogroups throughout the world. I'm going to come back to this. This is just a quick overview of where the haplogroups are throughout the world. And I'll come back to this later when I talk more about geography and mitochondrial DNA. But mitochondrial DNA, although it was worked on in the 70s, it really was the 1980s that really gave us mitochondrial DNA. I'm going to give you now a little brief history of how we came to work on it and where and how this all began. So this is sort of before my time. I was alive, but I wasn't quite studying this just yet. In the 1980s, a paper was published in Nature called Mitochondrial DNA and Human Evolution. And I always go back to this paper because really this is a paper that not only informed a lot about mitochondrial DNA, but it really informed a lot about anthropology in general. It was a landmark paper in my field because it really, really put on writing in hardcore evidence that we are an African species. And it showed through the mitochondrial DNA how we are all very closely related. We actually trace our ancestry to a woman we called mitochondrial Eve. This paper in 87 gave rise to the idea of an Eve, like the first woman, through the mitochondria, hence mitochondrial Eve. And I found a little image of a reconstruction there, and I used that image a couple of times. But this landmark paper from 1987 was published in Nature. It was actually just a couple pages long and then 1 or 2 diagrams on it. Even then, that's what the top level of publication. Nature Science, even in the 80s, were top notch. And 2 of the authors now are considered sort of the grandparents or so, the senior scholars of mitochondrial DNA. So Mark Stoneking is right there on the left. This is when he was in his 30s. He's now in his 70s. And Rebecca Kahn, who's retired and lives in Hawaii. She is actually my advisor's advisor. So I'm her grandson academically. And that's another thing. When I soon learned that, I felt really like attached to that molecule and to that stuff. Populations across the world, from sub-Saharan Africa, Eurasia, Australia, and Aboriginal New Guineans, and was doing something called restriction fragment length polymorphism, RFLIPs. And I'll talk about that in a second. And it was a very simple way, but it allowed you to identify different branches of a tree. It gave rise to the term mitochondrial Eve, and really was showing that a lot of the diversity in the world, or the deepest diversity that is found in Africa. But also outside of Africa, you have a lot of diversity, which is really a subset. Africa has the most diversity. The world has a diversity that is a subset of that found in Africa. So it really puts Africa at the root of the tree, at the base of the tree. And it showed that there must have been a movement out of Africa that led to the diversity that we see outside of that continent. They used RFLIPs or restriction fragment length polymorphisms. It was tedious. However, it really gave a pretty clear perspective. And you can see from that tree, that's actually looks like a horseshoe, but it's a tree with the ancestor A on the bottom right. And you see the deepest branches of the tree, the black circles going off on the right, and the rest of the tree going off on the left or the top, circling around with groups from Europe, Australia, Asia, New Guinea, and even Africa on the rest of the tree. So Africa had the deepest diversity as well as diversity mixed with the rest of the world. And that was sort of the key discovery there. And like I said, they were doing restriction fragment left polymorphisms, or RFLIPs. I'm not going to say that again. I'm just going to call them RFLPs or RFLIPs. And what this is you're not sequencing the whole DNA. You know what point, what mutation you're looking for, and you put an enzyme into your reaction that cuts at that mutation. So if you're looking at a mutation at position 10,000, then you look at a sequence that goes from the high 9,000s to the low 10,000s, and then you throw an enzyme into the reaction that'll cut at that position. And then what you do is you take your DNA and you run it through an agarose gel that separates bands into sizes. And if it actually separates, if it keeps it as a large band, it means that it did not have the mutation there, and it's still a quite long, 1,000 bases long. But if it cuts, all of a sudden you have 2 pieces of DNA. So it looks separated even on the gel. So you can see on the bottom right, or you can even see it when it looks at markers in autosomal DNA. If you don't have a cut, like you would see in the small a on the bottom left, you would have a large band at the top. But if you have a cut, in both cases, in big A, you would have a band at the bottom, and the heterozygotes would have both bands. But the idea here with mitochondria, if you don't cut, you have a large band at the top of the agarose gel, because agarose separates the bands by sizes. But if you do have a cut, you get a separation in the bands, and therefore that mutation is present. So you were really inferring mutations based on spaces, on separation on an agarose gel. Very tedious. I'm glad I wasn't doing this, but it was effective nonetheless. By the time I started doing this, we were doing Sanger sequencing, in which case you would use primers to extend parts of the DNA, and then you would run them through a sequencing reaction. And the reaction would actually color code lengths of the DNA with a terminal marker, the last one being highlighted, whether it's an A, a T, a C, or a G, in a certain color. The machine would then read the color and give you a base sequence read that would look like an electropherogram at the top. A sequence of different colors, where C's would be blue, T's would be red, G's would be black, A's would be green, and so forth. And then you would read the sequence across. And this is where I spent a lot of my time reading sequences. This is called Sanger sequences. This is what back in the early 2000s I was doing, but the Geographic project was doing. Family Tree DNA was doing this for many years as well. And this was where the field was in the early to mid 2000s. Now, by the 2010s, late 2000s and 2010s, we moved into a chip technology, which actually is still employed by many today, in which you would amplify the DNA and then you would put it across a chip where it would react either positively or negatively to probes on specific places of the DNA. And then it would light up in different colors, like you would see, green, yellow, red, depending on the presence or absence of that mutation. So in a sense, you already knew all the mutations you're looking for, and you could actually look at hundreds of thousands of mutations at the same time. And this would come out either as positive or negative for mutation, and then spit out a program which we turn into a spreadsheet, and would list my DNA, mutation position 1, I had an A. Position 15, I have a C. Position 500, I have a T, and it'll give you a long read like that. This is microarray or chip technology. And now we moved in the late 2000s and the 2010s, and now in the 2020s into next generation sequencing, which really combines several of these aspects. It is breaking apart long strands of DNA and making billions and billions of copies, and then forcing it to reconstruct, line up with each other, and then it gives you the best read. So you're really doing what we're doing sequencing, but hyper in a more at the millionth power in a sense with next generation sequencing. And this is what's being done today with some amazing machines that you see right there, the HiSeq. And at the end, what you want to get is your DNA sequence. You're looking for mutations. This right here is the hypervariable region. This is copied straight from a reference sequence. And when I was doing this, and people still do this now, they're looking specifically at certain places. Let's say 16,189, marked there in a little red arrow as a T. If you have a C there, that's often associated with haplogroup B in mitochondria. A couple of spaces down, 16,191 in the third column all the way in the right. If you happen to have a mutation, then you're haplogroup U or U5, like Paul. Or you might have other mutations in the 16,200, 16,300s, which are specific to certain haplogroups. And this is sort of what it would look like when you reconstruct those mutations in a tree. This is a tree down there of haplogroup H, which is one of the most common haplogroups in people of European ancestry. And for many while, we were calculating mutations in comparison to what was then called a Cambridge reference sequence. It's still called a Cambridge reference sequence. This was published in 81. So when Becky Kahn and Mark Stonking were working in 87, they were referencing everything against this Cambridge reference sequence, whose sequence was the first one to be completely done, the mitochondrial genome in 1981. People still use it today. We've actually moved more into what we call the reconstructed sapiens reference sequence, or RSRS. People realized that comparing your mitochondrial DNA, relatively derived, it actually ended up being haplogroup H2A and so forth, as it's sort of listed there. It was sort of backwards. If we could actually, now that we have been able to sequence thousands of people, if we can actually reconstruct what this mitochondrial Eve was, wouldn't that make a lot more sense and be comparing something to our shared common ancestor? And this is a paper published in 2012 by some great leaders in mitochondrial DNA, led by Doron Bahar, including other great researchers like Antonio Toroni and Tomas Kivisold and others, who got together and said, we have to reconstruct and not base everything off of the Cambridge reference sequence. Base it off on this mitochondrial Eve that we've been studying and know about for many, many years. This was a big breakthrough in mitochondrial DNA, the reconstructed sapiens reference sequence. And I believe it's the right way to do it, to compare to our shared common ancestors. But for many years, there was a debate as to how do we call this different haplogroups. If you remember that paper I just showed you in 1987, there weren't any labels. There weren't any haplogroups A, B, C, D, and so forth listed there. Stone, King, and Kahn were pretty much just grouping them by shared mutations, but there was no nomenclature. Fast forward to 1992, a group led by Antonio Tironi published a paper on Native American mitochondrial DNA. And in this chart, you can see where they immediately said, we're going to call this one Group A, call this one Group B, Group C, and Group D. They were studying Native American groups. They found 4 of them. Let's call them A, B, C, D. And that stuck. That became the beginning of the nomenclature. Almost by chance, this group published something and made sense. They called it A, B, C, D. And after 92, people started adding letters, and that's how it all began. Interestingly, my advisor published this paper on the bottom right in 1994. It was Dr. Koji Lam. He didn't use A, B, C, D. He used Roman numerals 1, 2, 3, 4, 5 to talk about Pacific Island haplogroups. And even when I met him in 2003, he was like, I can't believe these Roman numerals didn't stick. How come we're now using letters? So 10 years after Tironi and others had published that nomenclature, people still holding off. We're not going to go A, B, C, D. But by the mid-2000s, everybody had adapted the nomenclature of mitochondrial haplogroups, A, B, C, D, all the way through. We go through Z, and we now have subgroups as well of that. However, it was in 2002, really, with this publication of Brian Sykes publishing a book called $7 of Eve, that really started making this more and more popular. In the 90s, people were publishing, but nobody really knew what mitochondrial DNA haplogroup was. And this popular book, published in 2002, really brought mitochondrial DNA to everybody. He started Oxford Ancestors, a company based out of Britain, where you could test your own mitochondrial DNA. Shortly after, Family Tree DNA and National Geographic Genographic started working independently and then together on mitochondrial DNA as well. But this really was sort of a take-home, making mitochondrial DNA something that people could understand better by giving these 7 groups from Europe women names: Helena, Xenia, Ursula, Catherine, Tara, Jasmine, Velda. This is $7 of Eve, published by Dr. Brian Sykes in 2002. A couple more quick go through here about how do we take this DNA and turn it into a map? That's often a question I get, because we talk about all this DNA mutations and so forth, and then we put it on the map without really informing. I think most people would probably know this and understand it, but this is the way I usually explain it. If we were looking, instead of 16,000, if we were looking at 3 points of DNA in different parts of the world, and we look at the exact same 3 points in populations, in these 6 different populations, you happen to see it's CAT is the sequence in Africa, but by the time you get to Middle East or Central Asia, the same sequence is TAT, whereas in Northwest Europe it's AAT, or Northeastern Europe. In Northeastern Asia, it's TAG. The same one is TTT in the Pacific, and GAG in the Americas. So after that, we just play a game of connect the dots. We've been playing this since we were 3 years old. You get the numbers 1, 2, 3, 4, 4, and you connect all the dots and you make a unicorn. My daughter does this. So this is what we genetic anthropologists are doing. We're connecting the dots, but the dots of human history. By connecting, you can see one mutation transfer from here to there and another one from there to here. And obviously, this is not done by hand. This is done by computers, but this is sort of the breakdown of how we turn DNA into maps. We infer movement from one to the other and can trace that movement through the changes in the DNA. And now all that translates to this: big maps of haplogroups leaving at certain places, traveling out of Africa into Europe and Asia at different moments, evolving from L to M and so forth. Maybe familiar with maps like this. Really, it's the same thing as taking a mitochondrial DNA tree and putting it across a map of the world and color-coding it by continent like this does, and giving it sort of time frames as to when haplogroups first occur and where and how they evolve from one another. This is sort of, I jumped a lot in between the last slide and this one, but it sort of explains how we turn DNA into maps. We understand how it mutates. We understand sort of migration, how you move from one place to another, and infer movement, really, based on these mutations that make the tree. And this is sort of a comprehensive way to see both, right? This is sort of all of the haplogroups, all the major haplogroups put together here, color-coded by continent. Just quickly showing you with mitochondrial Eve all the way in the top left, L0, L1, some of the deepest branches. One branch down towards the bottom of that side evolves into M, another one into N, the 2 major ones outside of Africa. And you can see how different branches of M and N migrated across Eurasia even into Australia and the Americas. So this is really the map, the tree of mitochondria, and the map put together. hardly do we ever study everything in the world at the same time, right? So what now people have been doing is really doing much more localized migration, understanding migration within a part of the world by looking specifically at one or two branches that are sort of localized in that part of the world. Like if you're focusing in sub-Saharan Africa, you might be looking at L's and L's, L0, 1, 2, 3, 4, 5, and so forth, where they occur, where ancient DNA tells us they were 1,000 or 10,000 years ago, where they are now by testing living people. We can do the same thing in the Pacific. It's not just looking at migration from Southeast Asia to Australia or New Guinea. Now we can actually look at the sub-branches of P and Q, the major groups that you don't find anywhere else, but they're very common in New Guinea, for instance. Now with localized migration, we understand, P1, 2, 3 go this way, but P4 and 5 went that way. And then P9 and 10 end up in the Philippines. So it must have been something that happened even before they got to Oceania. One little cousin went off to the Philippines. We've been doing this for the Americas for about 20 years, 25 years, since Tironi and others were publishing there. Now you still see it. This paper on the bottom right is really hard to see, but it's a study in South America, a very specific branch of B2A1C, I think. Very specific. And we get that specific with full mitochondrial genomes, studying all 16,000 bases. We can actually see migrations in and out of the Andes Mountains with mitochondrial DNA. And another way to describe this is look at the haplogroups, where they're found. We put them on pie charts across the world. You've probably seen a map like this before. And really it shows you sort of centers of gravity, I call it, centers of concentration of certain haplogroups, A, B, C, D in the Americas with some ties back to Asia, P, Q, O in the Pacific, L's in Africa with L1s, L2s, L3s, primarily in this chart. But this is not showing you immediate migration. It's showing you sort of frequency, where their home is today, in a sense, haplogroups. But it's not just about academic publishing. We are a genealogy group meeting today here. And really, it was thanks to mostly Family Tree DNA and National Geographic that this became something that you could take home and do with citizen science and personalize it. So if you've taken a test with Family Tree DNA, you've taken one with Genographic, or even 23andMe does give you your mitochondrial DNA haplogroup, you can use that to sort of trace your own personal migration. Here's a heat map for haplogroup U. This comes from the website I used to work on in National Geographic, and you can map movements out of Africa into the Middle East and so forth of your ancestors from L0 to L1 to L2, 3, all the way to R to U, and then your sub-branch of U. You can see in the left side there. While I was working in National Geographic, we also did this by population. We went to Ireland and sampled hundreds of people in County Mayo in Ireland, and then gave them sort of a distribution of the mix of the population there, how many haplogroup U's and H's and J's and K's and T's and V's were there. And really knowing from our own studies where these haplogroups were coming from and when, we could really reconstruct the history of the population of County Mayo. We did something similar in Asturias in Spain. You can see on the bottom right. So we got some National Geographic geographers to help us map this as well. And people not only were personalizing it, but we could take this back to Ireland and present it to them and say, this is sort of the history of this county based on your mitochondrial DNA, or the mitochondrial DNA of 200, 300 people that participated. And it was very neat, a very neat way to sort of explain DNA to people. People wanted more sometimes, so people said, I haven't done this, but I own my own T-shirt. You can sequence your haplogroup and put it on a T-shirt. That doesn't get much personalized than that, or your coffee mug. So this is really, it is sort of hardcore science. It's sort of what genetic anthropologists have been doing for many years. But it really is something that people can personalize, take with them, because once you do your mitochondrial DNA, you can actually learn something about your own group, your clan, your tribe, in a sense. And you each get a letter or a subgroup and so forth, and put it on a T-shirt and display yourself to others. So I think I have my own. I didn't wear it today, unfortunately, but it's kind of a fun little thing to do. I'm going to pause here. So I'm going to stop my share. So that's the background on mitochondrial DNA, and I'll let Dr. Paul Maier take over.
All right. Thank you so much, Miguel. That was an awesome introduction. All right. So thank you so much. Thank you, first of all, to Tim for inviting us. And you guys, I'm really excited for this because this is like a great ragtag team of mitochondrial misfits here. Some of the greatest people on this subject are here. And I particularly love talking to genealogists because you are some of the most enthusiastic people with some of the best questions in my experience. So I'm really excited, and I think we'll cover a lot of ground today. A little background on me. So unlike Miguel, my background is not in human genetics. I worked on Yosemite toads for my PhD, and I've worked at Family Tree DNA for the last 3 years as their population geneticist. So more recently, I've been working on My Origins 3, on getting age estimates for the Y-DNA haplotree, and for this project, the Million Mito Project, and other mitochondrial-related projects. But back in grad school, I worked on Yosemite toads. So the nice thing about toads is that they're a lot simpler than human beings. Human beings, well, I guess toads, unlike them, they don't really care about privacy issues with DNA, which is nice. They follow fairly simple rules. So if you're a toad and you see another organism, if it's smaller than you should probably eat it. If it's bigger than you, probably run away. And if it's the same size, you should probably mate with it. And that pretty much defines toad living, which makes it very easy to study them. So for my PhD, I worked on understanding the phylogenetic history of their past, and then trying to understand something we call adaptive introgression, which is a topic we'll cover with humans later, kind of how they adapt based on gene flow, their current population structure in the present, and then predicting how they respond adaptively to climate change. I think that gives you an idea of sort of where I'm coming from. And, if I give more of a broad overview or more academically older context to things, that's why. So I'm looking more at evolution in general and the tree of life and giving that context, which I think could be helpful and give more perspective here. So let's give a quick overview of the origin and the function of the actual molecule, the mitochondrion. So mitochondria are essential for life as we know it, but they're kind of alien in origin. Virtually all animals have them. There are some exceptions. There are some single-cell parasites. Particularly, there was one in the news a few years ago, a cnidarian. Cnidarians are kind of jellyfish or corals or things like that. But this one is a salmon parasite that doesn't breathe at all, so it has no mitochondria. But that's pretty rare. Most animals need them, and mitochondria's benefit is that it gives the ability to mobilize energy in a very efficient way, and is pretty much the reason that animal life is possible. The other really strange thing about it is that it has a non-animal origin. So it's really just a proteobacterium that was engulfed a long time ago. So in the tree of life, there's kind of several branches at the root. Miguel kind of showed nicely how trees are structured and the roots of trees. So at the root, we have different groups of bacteria, and you can see that over time, mitochondria sort of reticulated, or they moved into the ancestor of plants, animals, and fungi. And the same sort of thing happened with plants with regard to chloroplasts or chlorophyll, which is how they're able to photosynthesize. But mitochondria sort of are a mutualistic relationship that has this old history. So again, this just shows the origin of mitochondria in the tree of life. You can see how there's sort of a gene flow event at the base of higher eukaryotic organisms like plants, animals, and fungi. And you can even see this phylogenetically, that if you look at the mitochondrial DNA, it has quite a lot in common with alpha proteobacteria, just like the chloroplasts in plants have a lot in common with photosynthetic cyanobacteria or aerobic cyanobacteria. So it's a mutualistic relationship because we need them. We wouldn't be able to persist without them. They're basically turning chemical energy into ATP, which is the sort of unit of energy that we're able to use and mobilize. So they turn all that sugar into this nice, delicious energy that we can use for all of our movement and everything we do. And at the same time, we offer them protection, and we offer them sort of an easy source of that glucose, that chemical energy that they're able to utilize. So it works for everyone. And because of that long-standing mutualistic relationship, they've lost most of their genome. So they are unable to persist on their own. So there are only 37 genes in the mitochondrial genome, and we'll get to that. And they include specific genes that have to do with what's called oxidative phosphorylation, or making food from glucose. But much of the genes that they actually need, or the proteins they actually need for their own transcription and replication, has been moved to the nuclear genome, to the nucleus. So the nucleus has about 20,000 genes, and about 1,200 to 1,500 nuclear proteins are necessary for mitochondrial to exist, but they're all in the nuclear genome. And because of this, there's kind of this back and forth adaptation going on, or this coevolution going on, where the nuclear mutations might sort of make an incompatibility in the mitochondrial genome and vice versa. And so you have this sort of tit for tat, stepwise process where there's an arms race and they have to be compatible, otherwise the organism suffers or might die. So there's a lot of adaptation going on between the two genomes. And this has been studied pretty extensively outside of humans. This copepods in California, there's kind of two different species in Santa Cruz and San Diego. And essentially they interbreed them and they look at the F1, which is the first generation after they hybridize, and then the F2, which is the second generation after they hybridize. And by the second generation, if there's incompatibilities, they break down and they're inviable. Mitochondrial DNA is circular because of that bacterial origin. It's double-stranded DNA. It's about 5 microns long, and so of course much smaller than the chromosomes found in the nucleus. They're found in human cells by the hundreds or thousands. So there are usually hundreds to sometimes over 10,000 molecules per cell. And each mitochondrion usually contains multiple copies, 2 to 10 mitogenomes. And there are multiple mitochondria per cell, and of course, trillions of cells in a human body. So we're talking about many, many copies of the mitogenome. There are some exceptions. For example, human oocytes, which are the precursors to egg cells, have hundreds of thousands. And as Miguel mentioned, it is inherited in a very specific and unique way. It's only matrilineally inherited, which means you only get it from your mother's mother's mothers, etc. This was first studied in the bread mold Neurospora crassa, but it's of course been shown in amphibians, mammals across the tree of life, humans, of course. There are some exceptions in the tree of life. For example, there is paternal inheritance in the saltwater mussel, Mytilus, but this is very rare. Exceedingly rare in humans. There have been a couple of cases of paternal inheritance. One of them is called into question, and certainly people who have this are not viable for very long. So it's not something that can persist. So it's matrilineally inherited basically across the board. And the way this happens is during fertilization of the egg and the sperm together to make a zygote, to make an embryo. The oocyte and the sperm each bring their mitochondria together, and shortly afterwards, the paternal mitochondria are degraded, and that can happen either passively, just by the fact that there are fewer of them, and also actively by essentially they get marked for extermination. Thinking of Miguel's matrix reference, they're kind of marked as a target and then they're annihilated by enzymes. So any delay in this process can really add to embryonic lethality. So it's done very efficiently. And so there are, of course, because there's so many mitochondria in each cell and mitogenomes, there are often more than one copy, and these are called heteroplasmies. They're quite rare to be in high frequency, but they are actually fairly common in very, very low frequency because mutations happen very commonly. It's a high mutating molecule. So due to this population of mitochondria per cell, you have this occasionally in a detectable manner. So you may have this in your results. It's certainly possible. Usually it's only above 60, 70% frequency where it can actually influence disease or anything like that. So it's thought to result in negative selection over multiple generations and be kept in lower frequencies where it has no effect. And there are different ways for keeping it lower. I'll relate to what's called genetic drift, which I'll get to in the next part of this talk. And basically through cells dividing, there's kind of a bottleneck where only a few mitochondria are selected into the next cell. And also within a cell, mitochondria are themselves replicating, right? So each time that happens, a subset of mitogenomes are selected. And so there can be drift over time in terms of which copies are copied. So let's take a closer look at the actual structure of it. So the human mitochondrial DNA was the first fully sequenced version of it at 16,569 base pair, at least for the reference. It encodes 37 genes. 2 of those are what are called ribosomal RNAs. The ribosomes are part of the eukaryotic cell and they're involved in making proteins. 22 of the genes are transfer RNAs or tRNAs, and then 13 of them actually code for proteins. They're protein coding. And as you can see on the right there, they're very tightly packed. So the nuclear genome is very different than this in that there's a lot of space. There's a lot of packing peanuts in there. There's tons of space between genes, and even within a gene, we have these things called introns that kind of space out the coding parts. Not the case with mitochondrial DNA. It's packed very tightly, and you can see the colored blobs there are the actual protein coding genes. They're colored the same way because they do similar things, and I'll show you that in a second. But they're all part of what I said before, which is this Krebs cycle or phosphorylative oxidative process of making energy. And so they're all related to that process. So if you look at sort of the different colors of these proteins on a cell surface, this is sort of how oxygen is used to make energy from glucose. It's sort of used to strip off protons, and that proton energy is what makes ATP, the energy source, so highly charged and energetic. tRNAs, I mentioned, there are 22 of them. What do? They basically convert the DNA sequence in triplets, so ATC, CCC, whatever, 3 sequences of nucleotides into an amino acid, which is the building block of a protein. So it's how you translate DNA into actual stuff, into actual protein coding stuff. And it's kind of interesting that because of their unique origins, mitochondria have a unique code. So there's sort of a general universal genetic code that applies to the nuclear genome, but it's different for the mitochondrial genome. And it's also interesting to note that if you look at the top there in the black bars, on the left, you see small black bars indicating for animals, the mitochondrial genome size is all about the same. It's about 16,000 base pair. But for plants and other organisms, it can be quite large in terms of hundreds of thousands or even millions of base pairs. So it's not necessarily conserved across the tree of life. Last couple of things to mention, the D loop is really used quite a bit and was used before we could sequence the entire molecule. So what is the D loop? Well, in Caltech in about 1971 or so, it was discovered that for an extended period of time, you could observe 3-stranded DNA, 3 strands. And this part of the molecule was sort of, we always rotate it to the top when we make these diagrams. What it was the beginning of replicating the mitogenome into a second mitogenome. So during replication, we have what's called the heavy strand and the light strand. And the heavy strand is basically where most of the genes are. It also has more G's than C's, but that's getting technical. So it has more genes, and when you start to replicate it, you sort of add this 3rd strand. That's kind of the beginning of making a new mitogenome. So they observed this, they saw that that part of the DNA, the 3rd strand, was displaced, so they called it the D loop, displaced loop. So that 1100 base pairs of control region, we also call it, is very useful because it's not conserved. It doesn't need to do anything in particular for many of the positions, and therefore mutations are observed more commonly. They're not selected against. And so it's much easier because of that faster mutation rate or that faster observed substitution rate. It's easier to make trees out of that. And so for a long time, that control region or D loop region was used, as Miguel mentioned, for constructing mitochondrial trees. And so for genealogy purposes, there are kind of 3 regions to this. There's what we call hypervariable or HVR1, HVR2, and HVR3. HVR2 and 3 are kind of the fastest mutating. And at Family Tree DNA, we actually combine them. We bundle them into sort of one package. The rest of the molecule, the coding region, we call it, is, it's the balance of it. It's 15,000 plus positions. So just to summarize here, we have 20 to 25,000 genes in the nuclear genome, only 37 mitochondrially. Most DNA chromosomes are linear, and mitochondrial chromosome is circular. The inheritance pattern is unique. Of course, as Miguel mentioned, mitochondrial DNA does not recombine. So it's inherited intact from one generation to the next generation, from mother to daughter and son. So although the nuclear genome, except for X and Y, X in some cases and Y is diploid, mitochondrial genome is haploid. Mutation rate is actually much faster in mitochondrial DNA, but actually we get more years per SNP, and we'll get to that later. So it's more time in between each mutation that we observe. And the segregation pattern is Mendelian for nuclear DNA, but it's sort of this homoplasmy versus heteroplasmy pattern in mitochondrial DNA. Okay, so that's kind of the overview of the molecule, what it does. So what I'd like to do now is give you guys kind of a more broad overview of population genetics. And this is a really fun topic. Forgive me for making this a little more, as I said, broad in nature, but I think this will really help later on when we're talking about haplogroups and how they're common in some places and less common in other places, and why the tree is sort of shaped the way it is. It'll really give a lot of context, I think, as well as heteroplasmies and adaptation and stuff like that. So I think this will be a good topic to set later topics up. My background is in evolutionary biology. The greatest idea ever is often said to be evolution by natural selection. And there are kind of 2 major insights. There's descent with modification, which you can see in the top right corner there. That's the tree, the first ever phylogenetic tree scribbled down in a notebook by Darwin on the HMS Beagle in the 1830s. And the second insight was evolution by natural selection. And the mechanism is basically parents overproduce offspring. There are too many to survive. Any differences among those offspring that's heritable is passed on. And those who have the inherited differences that are the most fit tend to survive. And thus you get change over time. That's natural selection in a nutshell. And there's plenty of evidence for evolution by natural selection. There's comparative morphology, as you see in the left there with the phylogeny of whales and whale relatives. There's comparative morphology and vestigial limbs, for example, in whales and snakes. You have comparative embryology, that embryos are more similar to each other when they're younger, and then ontogenetically over developmental time, they become more different. There's biogeographic evidence in the bottom right there, where groups of, for example, flightless birds are more similar when they're closer together, and the continental drift has sort of diverged them over time. So tons of evidence. Of course, we're interested in comparative DNA sequences, not anatomy. So in the 1920s, along came the population geneticists, and they reconciled heredity, which is Mendel, with evolutionary change in populations, which is Darwin and Wallace. And the secret was pretty complex math that we do not have to get into, but it's important to understand at a high level, because Darwin didn't have a problem with selection. He had a problem with heredity. He didn't know how inheritance worked. He didn't know how you could genetically get changes passed from one generation to the next. He believed that sort of all inheritance is blended, that mom's characters and dad's characters are blended together. But the problem is, over time, then we would all just be a mishmash of some intermediate state for every character. And of course, he had no access to DNA, didn't have a clue what that was. So what the population geneticists did was really important. And if you look at the bottom right, he's not using that guinea pig as an eraser. He bred guinea pigs to do experiments. There are two kind of epiphanies. The first is that there are discrete mutations, which we're all, I think, familiar with. So you can have something like brachydactyly, which is the shortness of the fingers and toes. It's a dominant mutation. So if you have any dominant allele, you'll have that trait. And you can do sort of a traditional Punnett square and predict what your children will have as far as that character based on what the two parents have. So that's easy. But what about complex continuous traits like skin color, height, etc.? So what they figured out is that it's the same concept. It's just that there are more genes involved, more genetic loci, more SNPs involved. So you could, if you wanted, make a Punnett square of 3 or 6 different locations in the genome and still predict the phenotype. And so it's more of a bell curve when it's a continuous trait because there are so many factors, but it's the same discrete principle. The other major epiphany was population thinking. So with individual thinking, you sort of ask, what would the children have based on the parents? With population thinking, it's, let's pretend everyone can throw their alleles, their gene copies, into a big jar. Let's say everyone in America, all 340 million, whatever it is, times 2, because there are 2 copies in each person, genes into a big jar. Let's picture them as like marbles of red and blue. And let's take some gene copies and move them into the next generation. And what does that next generation look like? And the question is, how much change is there from one generation to the next? That's population thinking. You could, in theory, still make a Punnett square with everyone's in America's gene copies or alleles, but it's sort of a different way of thinking about change. And so that brings us to the simplest ever model of evolution, which we call Hardy-Weinberg. Hardy-Weinberg is basically homeostasis. It's nothing interesting is happening. It's kind of the yardstick against which we measure what evolutionary change is occurring. So there are all sorts of interesting evolutionary changes. My favorite is genetic drift, and we'll get to that first, but there's also gene flow, there's mutation, of course, and then for nuclear DNA, there's recombination, and there's natural selection. Whereas most of these evolutionary forces only affect what we call allele frequency. That's, you can sort of ignore whether it's heterozygous or homozygous, how the 2 copies are paired in a person. For mating, if it's random mating or non-random mating, that actually affects the level of heterozygosity as well. So genetic drift, what is it? So getting back to our jar of beads or marbles, it's basically random sampling error. That's all it is. It's, you reach into a jar, you pull out gene copies, and you make the next generation of people. And just because you have a finite population, you're not going to get exactly the same frequencies as you had before. And it's not a big deal if you're talking about millions and millions of marbles, but if it's 10 marbles or 20 here, or I guess 10, then you're going to get potentially a very different frequency. So in this example, you go from frequency of 0.5 to 0.25 just by random chance. That's genetic drift, and it's really just, as I said, a sampling error that's exacerbated by small population size. So that's the key. Population size is smaller, as with a genetic bottleneck, as with moving humans to an island where there's a small founder population, that's what you get. You get more drift. And so what it does, it creates predictable randomness. It's unpredictable in a specific population, but across multiple populations, you can predict that you'll lose diversity, you'll lose gene copies. And that's what we see. So you get more distinction between populations randomly, and there's less diversity within populations. That's the effect of drift. And this led to some really cool discoveries. Like there were these fruit fly experiments done in the 1930s and 40s where they basically put fruit flies in different jars. And they saw exactly what I just said, that the fruit flies over time, they either fix for one gene copy or they fix for the other gene copy randomly. But the thing you can predict is that they'll all be fixed for something because they're going to lose diversity. The other cool thing is they noticed this weird effect where the actual size of the population, if it were 16, for example, the math did not work with 16. It only worked with a lower number, like 9. That's because of something called effective population size. Effective population size, basically it creates a lower effective size because not everyone in the population is passing on their genes. This happens if there's a size change, like global human population size has skyrocketed from 10 or 20,000, at one early point to almost 8 billion now. So we still have an effective population size of 20,000, but we have way more people. You get this also with uneven sex ratios because not every male, for example, is contributing to the gene pool. And how this is relevant, for example, to mitochondrial haplotypes and their frequencies, well, if you look at small populations, for example, the Coalescent or the Central African rainforest hunter-gatherers, you see high frequencies of very specific sub-haplogroups. For example, we have L0D and L1C in the Coalescent and the Western rainforest hunter-gatherers, respectively. So this is because essentially they've lost some of those lineages, many of those lineages over time due to drift. Gene flow is kind of the opposite of drift. So whereas drift removes variation, gene flow restores it. It homogenizes populations. It makes them more similar to each other. And so, this is relevant for haplogroups again, because it's the reason that many times haplogroups don't correspond perfectly, or sometimes at all, to populations, because you can get one migrant moving between populations that might be a maternal ancestor of a whole subclade of a haplogroup. And therefore, you get this decoupling between haplogroups and populations. And of course, it doesn't take many migrants, but if you have many, it exacerbates that. This is also interesting to consider how gene flow and drift affect trees and how trees don't necessarily reflect the actual history. So if you look at humans and chimps, they are sort of sister species, and the outgroup to humans and chimps is gorillas. But for 25% of our genome, we get a different tree for our nuclear genome, that is. So for 25% of our genome, actually, chimps and gorillas are sister taxa. And for part of it, it's gorillas and humans are sister taxa. And that's because of drift and mostly drift in this case. Drift, basically, because you have this ancestral polymorphism, you have all these gene variants before those three species split apart, those gene copies can randomly sort in a way that doesn't reflect the actual history. So that's drift. Of course, if you did have gene flow in some other example, that could also cause the phylogeny to appear differently than the actual history. Natural selection is a big topic we're not really going to go into, but here's just one example with poison dart frogs in Panama, where there are these different islands with different colored poison dart frogs, and they've evolved different toxins. And because of their toxins, they have to evolve different autoimmunity. They have to become immune to their own toxins. And so if you make a gene tree of just that toxin-related protein, it sorts in that way. So you have adaptive evolution due to natural selection. In mitochondrial DNA, again, this is an example where the nuclear genome and the mitochondrial genome are co-evolving. They're adapting to each other. So as you have one deleterious or harmful mutation in the mitochondrial DNA, you might have to have a compensatory mutation in the nuclear genome, and that could be a sort of tit-for-tat co-evolving process over time. So in summary, drift reduces variation inside of the population, but it increases differences between them. Gene flow does the opposite. Mutation increases diversity, period, across the board. And that natural selection is pretty complicated. So how do we study this in PopGen? Well, we can either predict evolution through time in the future, doing simulations, or we can use gene trees, like the mitochondrial gene tree, for example, to sort of explain our history, our past. So we call this coalescent theory. Imagine we have 5 people. We'll say that this is a diploid locus. So it has 2 copies. So 5 people, 10 gene copies. And let's say we have genetic drift operating here. So in generation 2, what's the probability that people have the same copy from one parent? It's going to be 1/10. And if we extend this forward, you can imagine the top is mitochondrial Eve, for example. Over time, there's only going to be one ancestor to the entire population of people, or perhaps mitochondrial haplotypes. And so if we look at just what's left today, and we sample, because we can't sequence everyone, we sample 5 haplotypes, we can build a gene genealogy. And the time to coalescence depends in part on population size. So this is drift again. Genetic drift causes there to be a relationship between the population size and how likely there is to be a coalescent event in the past for 2 branches to come together. So why do we care? Well, because we can detect these changes, for example, bottlenecks, and we can do some really cool stuff. We can detect what happened, demographically speaking. We can detect if there was a declining or an expanding population. We can simulate sort of many random genealogies under a model, and we can determine which of those genealogies fits the observed DNA data pattern the best. And we can decide, was there population growth, bottleneck, was there gene flow or admixture, was there simple divergence, and so forth. And we've done this multiple times. So here's an example where we have looked at mitochondrial diversity through time, and we've reconstructed sort of a growth rate of mitochondrial haplogroups over time and found that there was a big expansion event just before out of Africa, or perhaps just after out of Africa, around 50 to 60,000 years ago. And this may correspond to the out of Africa event itself, could correspond also to demographic expansion before that set it up. So we can figure out really interesting things about our history just using coalescent theory, the principles of drift, and mitochondrial haplotypes. So I hope that wasn't too painful and too off topic, but I think this will really set up some of the other topics that we go over. And when we're talking about everything from phylogeography to heteroplasmies to how to use genealogical information, I just want you to think about this in terms of a coalescent process. Haplotypes are all related to each other and have a common ancestor if you go far enough back, and we can infer patterns, we can infer processes from those patterns and figure out what happened back through time. So that's the last of PopGen. And my last topic before we go to the next part that Miguel is going to talk about is phylogenetics. In the early days, in the 1800s, we didn't really have a concept of common ancestry. We just organized things as domains, kingdoms, classes, orders, families, etc. And that was Carolus Linnaeus in the 1700s. Our early concept of evolution was a progression. So bacteria evolved multiple times and they progress to better and better states. Eventually they get from fish to frogs to birds. After that, there were people like Ernst Haeckel who thought, no, there's change and it's one tree, but it's progression from bad to better. It's sort of a progression from early states to more derived and better states. Darwin's contribution was we have common ancestry and there's no progression to it. There's no goal. There's just common ancestry. So we had the early systematists, and after Darwin, we had this concept of branching trees of life. So there are a few times in scientific history we can say something's absolutely right or wrong, but this is kind of one of them. It's nice to have all the information, not do any of the work ourselves and look in retrospect and say, okay, we got it wrong before Darwin. So after Darwin, how do we actually build trees? So does one similar character mean necessarily that there's common ancestry? Well, we can think of cases where this is not the case. With pterosaurs, which would be like pterodactyls, birds and bats, they all share wings, but they're not actually each other's closest relatives. And you can think of the same sort of thing with homoplasy, which we'll talk about later. Those are characters that evolve more than once. So eventually over time, we build these large phylogenies. And phylogenies are just clusters of taxa. They can be species, they can be populations, they can be haplotypes. And they have two kind of attributes to them. One is that they're clusters of similar attributes. And the other is that those clusters are hierarchical. So you have a subclade, that subclade is in a larger clade, that clade might be in a haplogroup, and that haplogroup is in a macrohaplogroup. It's important to note that because we're genealogists, we always think in terms of pedigrees, but pedigrees and phylogenies are two different things, and pedigrees kind of are unique in that they expand both forwards in time and backwards in time. So if you look at yourself, you have 2 to the n ancestors back in time, and potentially 2 to the n descendants if you have the same expansion rate going forward. Whereas phylogenies, they only expand forwards in time. I didn't have time to change this from a Y-DNA version, but the concept is the same. So let's just pretend that these are females and not males that I've labeled. As Miguel set up before, mitochondria are inherited matrilineally. And so if you have a distant cousin, you might match that person. And the idea behind the haplotree or the phylogeny of those sequences is that you share a most recent common ancestor at some point back in time. So the question is when that was and what traits that haplogroup shares. So it could be very far back in time. I mean, if you're in haplogroup J and I'm in haplogroup L0, it could be mitochondrial Eve is our most common ancestor, but it could be something much more recent. Let's just imagine that these are not nuclear chromosomes, but rather mitochondrial chromosomes. And so if they were nuclear, over time, the blue segments would get smaller and smaller. The matching segments would get smaller and smaller due to recombination. But because this is shared mitochondrial DNA, there's no recombination. And so the only differences are mutations, just SNPs and indels and heteroplasmies. So that makes it very easy to infer a common ancestor. So that's how we're able to go from what you see on the left, sort of a pedigree of matrilineally shared lines, to a phylogeny or a haplotree. That's basically what we do in phylogenetics. We infer phylogenetic relationships from DNA sequences or other traits, and then we map those traits onto the tree and we try to infer the evolutionary origins or the events or the timing. So let's go over a little bit of terminology so we're all on the same page. A node is just a point in the tree that diverges. Internal branches are any branch that doesn't end with a sequence, so internal to the tree. Terminal branches we call leaves sometimes, so those are the external branches. And then the root, there doesn't have to be a root. So a root is something we add to the tree if we have that information. And the root is just a branch that's at the sort of beginning of the progression of the tree's evolution. So that's mitochondrial Eve in the case of mitochondrial DNA. Quick quiz, how many unique trees do you see? So the answer should be 1. We could add another tree here to make 2 unique trees, but the 4 on the top are all the same. They're just rotated versions of the same tree. 4 and 3 are sister taxa, 2 is the sister taxon to that, and then 1 is the outgroup. So the idea is that you can rotate nodes in a tree and it's still the same tree, it's just a different way of showing it. I should just probably also mention that there are different ways of displaying phylogenetic trees. You can have this sort of square bracketed version, or you can have diagonal lines. Sometimes, like Miguel showed, you can have kind of a horseshoe or a circle shape. So they're all ways of displaying phylogenetic trees. So what are we trying to find in a phylogenetic tree? What are we trying to optimize? Well, we want to find clades. We want to find haplogroups. We want to find monophyletic groups. Monophyletic groups are simply ancestors and all of their descendants. Ancestors and all of their descendants. So everything colored in here, green, blue, yellow, and red, those are all monophyletic groups, or clades or haplogroups potentially. So an example of this might be L3, because L3 includes all of its descendants, including M and N, the 2 out of Africa subclades. But there are paraphyletic groups, which are ancestors and not all of the descendants. So if you look at the far right, we have the ancestor A, we have 2 descendants, J and K, but we don't have descendants D through H. So that's paraphyletic. So an example of this in the tree would be haplogroup L. L originally described all of the diversity in Africa, but of course it doesn't include out of Africa. So it doesn't include subclades M and N, and therefore it's paraphyletic. Sister taxa are just the adjacent branches in a tree. So in the example here, placental mammals and marsupial mammals are sister taxa. Outgroups are just the sister taxon that's the farthest out. So it's squamates or reptiles in this example. And polytomies, you can see on the right-hand side, polytomies are just unresolved branches where you get more than 2 taxa that are next to each other. So usually this indicates uncertainty in how those are related, or they're quickly branching, so it's not enough markers to split them apart. Every unrooted tree also has multiple rooted trees. That's because the root itself is just a branch, and hypothetically, you could put the branch anywhere in the tree. Of course, there are not equally likely places it belongs, but it could go in any place between any 2 nodes. So that's why there are multiple rooted trees. Miguel's going to mention homoplasy, so I won't say too much, but essentially homoplasy is when a character or a trait has the same form or function, looks the same, but it evolved multiple times independently. And then we have these terms called apomorphy is just a character, okay? Apomorphy is just a character. So autapomorphy is just a unique character. It's only had by a single sequence or a branch. So we call these private mutations usually. Synapomorphy means a shared character, so it's unique to an entire clade, or it's a branch-defining variant. Symplesiomorphy is also shared, but it's ancestral. It's not shared recently. And then, like I said, homoplasy is shared through independent evolutionary events, so it actually doesn't cluster 2 branches together. So here's an example in the actual tree. So if we focus in on L3E, which is in red, with respect to L3E, that mutation at the top, 10,819, is symplesiomorphic. It's shared, but it's ancestral. So not just L3E has it, but also L3I and L3X. But those 2 mutations I'm pointing to below that, 2352 and 14212, are synapomorphies because they're shared only by L3E and they're defining. Underneath that, if we hypothetically imagine that 14 at the bottom is just 1 sequence, then that would be a private variant or private variance. And on the left, we see recurrent SNPs. 14905 is shared between 10 and 14, and therefore it's homoplastic or it's recurring. So we can take a few examples on the actual tree. So this is mutation 3666, and you can see that it's shared by everything downstream of haplogroup L1, and it only evolved once. So in red is the derived state of A, and in black is the ancestral state of G. Pretty simple. Here's a slightly more complex example. So 7055, the ancestral state was A, and the derived state is G, but you can see how it evolved more than once. It evolved first for L1, and then there was a reversal event for L1C3, and we have a notation for that where we, of course, have to flip ancestral and derived now that G is ancestral and A is derived. And we add an exclamation point afterwards, which denotes that it's a reversal event. So first A to G, then G to A. We have many recurrent SNPs. And like I said before, some regions of the mitochondrial genome mutate much faster. So 150 is in the D loop. And so we see here 4 independent evolutions of C150T. And sometimes we have what are called transversions. So transitions and transversions are just which base turns into which other base. Transitions happen much more commonly than transversions, and that's why we distinguish them. So it has to do basically with the actual physics of the DNA molecule, and we have purines and pyrimidines, and they're sort of bigger and smaller, and so it's much more difficult to fit in a larger nucleotide into a smaller space, so it happens less frequently. When we have a transversion, we use a lowercase letter like this, T7624 lowercase a for haplogroup L2BC. And then we have what we call indels, which are not mutations at one site, but they're insertions or deletions of multiple nucleotides. So here we have the insertion of multiple C nucleotides at position 5899. And this is defining for L1C24. That haplogroup in Western Africa has multiple Cs. It's sometimes a single one, sometimes 2, sometimes 5 or more. And because it's uncertain the exact number of Cs, we put an X there. If it were simply an insertion of 1C, it would be 5899.1C. And same thing for deletions. We can have a deletion of, in this case, 8281 to 8289, and that occurs in L0A2. So that deletion is denoted by 8281-8289D for deletion. Roberta is going to talk more about genealogy later, but she has this fantastic example where she's looked at her own private mutations, which are shown in yellow at the top. So those are what are called extra mutations, which means they're private to her. They're not on the tree. So below that, she has people who match her, but have a genetic distance of more than 0. So they have some mutations that are not shared. And so one thing that our working group is trying to do in the Million Mito Project is we're going to add these new mutations that are not on the current tree and add them to the tree, build a larger tree, add more clades and subclades. So in the future, this sort of exercise shouldn't be necessary, but for now, it's interesting to look at your matches who are not perfect matches. They're not GD of 0, but they're genetic distance of 1 or more, and see what mutations they share and try to figure out, okay, do they fit into their own clades and subclades, and how does that relate to my clade? All right, great. That's all nice, but how do we actually build trees? So let's take a quick example. What we do is we build a list of characters or DNA positions, and we figure out which ones are variable, which ones actually change across different people. We get a list of samples or haplotypes, and we make a matrix of sequences by characters. And then we basically have to look at every single possible tree that you could build with those sequences and figure out how they fit onto the tree. So how many trees are there? Well, there's a lot. So for only 4 taxa, there's 3 possible unrooted trees. As you can see, it becomes very quickly unviable. The quote is more than 53 species or 53 taxa trees have more trees than there are atoms in the entire universe. So that's not something we can feasibly do. We can't look at every single tree. So in that case, we have to sample the trees. We have to look at the good trees, if you will. So the question is, what constitutes a good tree, and how do we find it from all the possible trees? Well, today we're not going to go too in-depth in this topic. We're going to keep it high level, but a method that's been used a lot in the past is called maximum parsimony. So we'll use this because it's a good teaching example. Just note that we don't really use this anymore for various reasons. And then how do we find the best tree from all possible trees? Well, you have to sort of sample the different trees in an intelligent way. So there are ways of doing this where we basically take branches of the tree, we separate them from the tree, and then we graft them to another part of the tree. And we do this in an intelligent way where we're trying to optimize good trees. We're not sampling bad trees. And there are ways of building algorithms that can do this well. All right, so let's make a maximum parsimony tree. We have our characters, we have these 5 different nucleotide positions, we have our haplotypes, haplotypes 1 through 4, we have our matrix shown here in color. So we have to find from all possible trees the best tree that supports the data. How many trees? Well, in this case, like we said, there's only 3 possible trees. Okay, so which characters are informative? Well, if you look at characters 4 and 5, you can see that they don't group anything together. Character 5 is the same for everyone. Character 4 is only has a private mutation for 1 sequence. So it can't group 2 sequences together. So we discard those 2 characters. So that leaves us characters 1 through 3 to map onto trees 1 through 3. So we'll map these out. So we have our 3 trees, we have our 3 characters, and then we can look at each one and add mutations. So we can add for the first character, we have AA, GG. On the first tree, there's only 1 change because AA and GG are in different clades. On the other 2 trees, you have to add 2 different changes because of the way the trees are structured and which mutations those sequences have. So there's 2 changes for each of those. Character 2, only 1 change, 2 changes, and 2 changes. And character 3 is 2 changes, 1 change, and 2 changes. So if you look at all of these trees, which one is the best one? Well, based on maximum parsimony, you're trying to limit the number of changes because you think that's the most reasonable tree, right? There should be fewer changes through time. That's more parsimonious. So you choose the tree on the left because it only has 4 changes, whereas trees 2 and 3 have 5 and 6 changes. So that's the best one. And we're not going to get into sort of how we decide what a good supported clade is, but there are ways of deciding which are the best clades and which clades we can collapse because they have not enough support. All right, so with that, I know that's a lot to take in. I'm going to hand it back over to Miguel, and I think he's going to talk about homoplasy.
Thank you, Paul. That was great. I think we're almost at 90 minutes, so I think maybe I'll just talk briefly about the networks and homoplasy that you introduced, and then take a break, or there are chances to go a little further. So I'll leave it up to Tim after I speak briefly, but I only have a few slides. I'll share my screen again. But thank you, Paul. That was fantastic. That was so well explained, and actually leads nicely to what I have here. Now, Paul was just explaining so well how to make trees and how do we deal with some of the situations like homoplasy. And I'm going to go into that more in depth because trees are fantastic, and trees are just a really good way to show phylogenetic relationships. Another one, which is very similar to a tree, is a network. And I'm going to talk about how those are being used and some of the benefits of networks as opposed to trees. And I'm showing you sort of side by side. On the top left there is a tree. That is what actually, when you open up a phylo tree, very commonly used for mitochondrial DNA research, it's a tree right there. It really is sort of a root at the top, upside down tree with all the haplogroups at the bottom with bifurcating branches. Which is not necessarily always the case, but it is with those monophyletic groups that Paul explained so well in haplogroups L0 through L6. But then you get to L3, and then you get at least 2, possibly 3 different daughters of L3, right? L3 star, or maybe that's actually more than just one L3, but M and N. So then all of a sudden you get multiple points of bifurcation. You get trifurcations. You get several different descendants coming from one ancestor, which can get a little messy sometimes in trees, but it's doable. I argue sometimes that networks could be better. So networks is what we have shown to the right there. Top right is a very colorful network showing you various different haplogroups. And usually what they are just sort of a large node, a large circle, and the size of the circle represents the number of individuals that share that haplotype, share those mutations. So instead of putting the mutations down, you would put a circle, and the size of the circle could be very small, 1 individual, to very large, say 100 individuals. That would vary. You would have to put that in your explanation. And then all the circles are connected by branches, and the branches represent the mutations that separate the individuals or the lineages, the haplotypes. So the bottom shot there is a nice sort of depiction of a network used in mitochondria. And why I like it so much as well is because you can actually overlay pie charts on it very nicely. So it's double informative. The pie chart shows you how in the percentages of that node, that big circle, is shared across different populations. Here you can see a haplogroup shared across all these different Mediterranean populations from Italy going all the way to Lebanon and Libya. And then the branches very nicely show you the mutational spots. So quickly you can infer so much. You can learn so much from a network like this. You can see network, what haplogroup it is, but that's not important right now. The importance is to show how the nodes show size. They can show diversity across different regions of the world or different parts of the Mediterranean in this case. And then the mutations have the list, the branches have the list of the mutations right on them. So really, if we go back to the same beautiful tree drawn by Darwin and while he was traveling in the Beagle, and I think, Paul, you used the same tree 2 or 3 times. I'm bringing it right back because he's sort of he established this. He's the great grandfather to us all here when it comes to population genetics, although he didn't know about that mitochondrial DNA. But what he was doing there is really a tree, but it's not that different to a network, and that's the argument I'm making here. You could sort of see what he was doing was sort of showing how different groups, and this, I guess this is different species or the origin of species, A, B, C, D, how they're related to a common ancestor 1 on his backhand tree that he drew. Well, when you put that as an actual tree, you have to sort of have nodes of connecting common ancestor between A and B will be that node there. Common ancestor between A, B, C, D, E would be that one there in a root. But that doesn't necessarily always represent well the phylogenetic relationship. Whereas a network, which is actually not that different than what he drew. I kind of drew a network. He just wasn't putting the mutations or the nodes in. What he drew is actually sort of the grandfather both of trees and networks. You'll be able to put the colors, the size, the population level of each node, and list the mutations as well. And here's one of haplogroup M with the root in the middle. And right there, you can see certain mutations that lead off of that root of M towards different subgroups like M49 on the right and M4B on the left, and where they are in various places of Asia. I think that they're quite informative. And they also solve that problem with homoplasy. But before I go in there, I want to show you this. And this is not mitochondria. This is COVID. This is a network right now that we should all be aware of, but we should understand well. This is what the best scientists are doing right now to save us from this pandemic that we're in. They're understanding where different viral DNA originated and how it could have spread. And they're using networks to explain this. So on the top right is one of how the original one in China spread to other parts of China and in the Middle East, but one makes it to Australia, Sydney, Australia, eventually makes it to Italy. Remember last spring, we had a huge peak in Italy, and we were worried about China coming. And yes, that yellow does migrate, make it into the west coast of the US, but later found out because of networks, we found out that it was actually a European COVID that came over and really hit the United States more heavily. The bottom left is another sort of representation. I've seen this a lot, and I'm very excited to see the success of networks explaining the diversity and the spread of COVID. But what networks can do that trees can't do very well is this homoplasy situation. Again, we use each other's charts, it seems, Paul. I'm using the same one you use with the homoplasy there at the bottom right, explaining what it is. And it's when you get a derived trait, or in this case, a derived mutation occurring twice or different places of the tree. And if you're just having a computer simulation run this for you, it may not necessarily even see the mutation if you have it go forward or backwards, or it may not know what to do when you see the exact same mutation in different parts of the tree. But if you do it as a network, you can have this represented in different types of reticulation. You can see a branch coming one direction leading the mutation A, and another mutation leading to the right, mutation B. And then one individual maybe has both A and B. You don't know if you got A first and B second, or B first and A second. So instead of drawing this as different branches and trying to computer calculate, or just you guessing A was the first mutation, B was the second mutation, you form a rectangular reticulation in a sense. In this case, it's shaped like parallelograms there on the network, which sometimes is not that informative because you still don't know which one occurred first, but it's actually the real representation of the data. Because you can't sometimes tell which one occurred first until you get maybe ancient DNA, or until you get something else that really shows which mutation occurred first and which mutation occurred second. And it's not always easy to figure it out. So networks can alleviate this, can cure for the situation of homoplasys. Another sort of example here, what would happen when you get a sequence of, I'm talking about right there at the bottom center, TTATCC, right? And you get in one branch a mutation right there in position 2 that changes the T to a G. But a separate branch actually changes the same mutation from a T to a C. Well, what if that G had also mutated into a C, and we're only seeing the outcome? We're only seeing TCATCC, and we refer the two bottom ones are more closely related to each other because they both have a C. And we couldn't tell that actually the G was an intermediate step before getting to the C. That's creating sort of an extra step. The tree, they're corrected for that because we were able to infer that direction, that this mutation C occurred in two different branches, but it's not always quite obvious with a tree. This is sort of another example. Sometimes networks are quite simple, quite easy to show sort of a nice large center with sometimes dozens, if not hundreds of branches coming off, like we see in haplogroup H, the most common one. You have an ancestral H, and then you have dozens and dozens of sub-branches of H. That's shown very nicely in the network. The network, again, can show these reticulations that would explain homoplasys, as you can see very well on the left side. So again, homoplasys create a problem for us when we see them in DNA, but networks allows us a way to sort of explain these better and more correctly. So anyway, I wanted to put a plug there for the use of networks together with trees. And when I go in the second half of this, I'm going to talk about academic research in mitochondrial DNA. You'll see how at least in the 2000s and early 2010s, network was sort of the number one way that we were presenting mtDNA diversity throughout the world. So I think that's my last slide.
All right, so the next topic is going to be about the molecular clock. In phylogenetics, there are sort of three different forms of a phylogeny. You can have a cladogram on the left, which only shows the relationships between different taxa or sequences. The branch lengths don't actually mean anything. They're just the relationships and nothing else. You can add in lengths, which could just be the number of SNPs, could be number of indels, number of characters. So we call that a phylogram. And then if we actually scale it to time, we call that a chronogram, chrono for time. So if we want to find the time to most recent common ancestor, we have to create a chronogram. That's the goal. So we already showed how genealogies or matrilineal inheritance patterns mapped onto genealogical trees kind of have most recent common ancestors embedded into the idea. And so we can create haplotrees based on those patterns. And the idea of making time to most recent common ancestor is that the age of the most recent common ancestor, or the MRCA, is also the age of the genealogical MRCA. So how do we figure out that timing? Well, we basically have to smooth out lots of variation in how often mutations occur. So in a phylogram on the left, you can see that there's a ton of differences in how long each branch is based on the number of SNPs, number of indels, and we have to smooth that out. This is, again, just kind of showing what we'd like to have for the mitochondrial tree. We have this ultrametric or chronogram tree showing every single leaf of the tree ending in the present time. And therefore, every single node, every single ancestor has a time associated with it. And then we can do cool things like map on the autosomal origins and figure out how different haplogroups relate to different populations. Mitochondrial age estimates are a little more complex. So it would seem simple enough. The mutation rate is about 2.285 times 10 to the 8th, or minus 8th rather, sites per year. So that corresponds to about 0.02 substitutions per base per million years. And that's roughly 30 times faster than the Y chromosome. But because the mitochondrial genome has 600 times fewer sites than the Y chromosome, it yields about 20x slower years per SNP. So it's a smaller molecule, and therefore that reduction in size means that there's more time between individual SNPs that we observe. So that seems simple enough. We can just use a clock like that. However, there's massive variation that we have to account for. There's site rate variation. So because you have genes involved, genes are under natural selection, which we talked about. So you have these, remember, codon positions, these triplets of nucleotides. It can be CAT, and that cat can encode a certain peptide. And so not all of those three positions are under the same amount of selection, because position 3 can evolve much faster without any effect downstream to the protein. There are pressures that constrain how fast different sites can evolve. So there's codon position variation. There's also non-codon variation. We talked about transitions and transversions. Each lineage potentially could evolve at a different rate if there's a clock rate shift. And then there's all sorts of positive and negative natural selection pressures, as well as mutation rate that we observe in a genealogy could actually slow down in deep time because we're not observing all the mutations as they exit the population. So these are all things, in other words, that we have to take into account. What this shows you is just one of those types of variations. So the years per SNP is shown at the top for different types of positions. So we have codon position 2 at the top there, evolves the slowest, about 5,300 years per SNP. And the average is something like 2,600 years per SNP. And at the very right side, we have the hypervariable region, which evolves much faster at about 450, 460 years per SNP. So this is all stuff that we have to take into account when using a so-called molecular clock. This is showing transition to transversion ratio. So like I said before, transitions happen much more frequently. Transversions are very rare. So they happen in different ratios in different types of positions. In position 1, it's about 50 transitions for every transversion. For the hypervariable region, it's about 20 transitions for every transversion. So another type of variation that has to be taken into account. And then, like I said, mutations in shallow time and recent time can seem very fast, and this has been calculated for pedigrees. But the deep time substitution rate, we call it, can be much slower, and so we have to account for that in some way too. And then finally, like I said, each lineage, each haplogroup might have its own molecular clock. This seems not to be a major issue, but it could be an issue in some recent subclades, especially ones that have had demographic increase. We talked about population size increases, so that can affect the molecular clock in subclades as well. Mitochondrial Eve is simply the ancestor to all extant mitochondrial sequences today. But of course, mitochondrial Eve will change over time as the tree changes and lineages go extinct. It'll be a new Eve more recently in time as we move forward. So it's just sort of a fact of there being one ancestor to everyone who's still alive today, every mitochondrial sequence that's still around today. So currently the most recent common ancestor of Eve, which is the branching between L0 and the sister branch, which is L1, 2, 3, 4, 5, 6, is about 145, 150,000 years before present. And it's kind of interesting to note that even though Eve, that one woman in Africa, is the ancestor to every sequence that's around today, her cousins, her family members, may have descendants just in the nuclear genome as well. So it's just a fact of how haplotrees are structured and the fact that there has to be one ancestor for each locus. And just one example here about haplogroups and where mitochondrial Eve may have been. L0 is very frequent in Central and especially Southern Africa. L0D and L0K, as I mentioned before, are very common in the Khoisan people in Southern Africa. L1C is very common in Central Africa in the Western rainforest hunter-gatherers. So where Eve may have lived is still debated, but probably somewhere in Southeastern Africa, which I think Miguel is going to talk about in his next portion.
So I'm just going to cover quickly, basically the use of phylogeography, really, of mitochondrial DNA, how we do this. I'll touch briefly, like Paul said, on how did we pinpoint mitochondrial Eve and whether or not we can truly believe that or whether it's just our sort of fantasy of wanting to find a person or a place or a location, or really the thought of it being this person. I'm a big fan of maps that sort of explain things both phylogenetically and also geographically. This one, borrowing from Tumas Kivisild, also a great mitochondrial DNA scholar himself, really shows the diversity of the haplogroups, and really at a haplogroup level. We're not even diving into the subgroups here, how it's spread, how all the different lineages are connected. And it really sort of, if you look at it deeply, shows you how humans progress from Africa to the Middle East and in Asia, in Europe, Australia, and Americas, and so stage step-by-step basis. So as an anthropologist, here's where I spend my time, sort of diving in, and it's like a kid in a candy store. But what I want to move on to from here is more this concept, and then move on to more in phylogeography and academia as well. So yeah, we've all been fascinated with the idea of mitochondrial Eve since the late 80s and early 90s. And like Paul has suggested, this is sort of somebody that we place, just a fact of phylogenetics, that it coalesces to one person who lived about 145, 150,000 years ago. We're assuming it's Africa to our best understanding based on research and based on where the deepest lineages, namely L0 and L1, and maybe even some sub-branches of L0, like L0K and D, which are primarily found in Click-speaking groups, the Khoisan-speaking in southern parts of Africa, as well as a couple places moving up the east coast of Africa towards Tanzania. So a couple of years ago, scholars and both science writers and sort of popular media got behind this and tried to place mitochondrial Eve somewhere. And us scientists were kind of hesitant, saying, you really can't do this. This is sort of a moving target, first of all, like Paul pointed out. But it's also, to the best of our knowledge, it's an estimate. But the reconstruction would suggest that we're talking about somewhere in southeastern, maybe south of southeastern Africa. I believe it was named either Zimbabwe or Botswana as a place, and both countries took to a point for tourism and promotion. They were even pointing it down to a certain area, a certain village, and so forth. It's kind of neat because this is sort of the interplay between like hard science and popular culture. And I think as more sequences come out, and as we reevaluate sequences that are there, we'll talk about that at the end. We'll talk about the Million Mito Project. We'll see that it's a moving target. And that's what I love about anthropology. Really, it's gray. It's not black and white. And that's what makes it so enticing to come back every day and ask newer and newer questions. So I think what fascinates me most is sort of this localized migration. So we've all seen those maps. We even, as Genographic, we gave people a map when they bought a kit that showed the migrations and the haplogroups of the world. But really, for the next 15 years after that, really, it was all about localized migration, understanding sort of movement of certain subgroups in certain parts of the world. I showed this slide earlier as well. And that still continues. But really, there's some major changes that were happening in the 2000s that really changed genetic anthropology, maybe away from mitochondrial DNA. And so in the 80s, with the first paper, Anderson and his group, later with Becky Kahn and Mark Stonking and Alan Wilson in 87. And then I mentioned the Taroni group, including Doug Wallace, Tad Shore, a former advisor of mine as well, working in Native American populations. We were seeing sort of this progress, and mitochondrial DNA was the dominant thing to study in the 80s and 90s into the 2000s, probably peaking with Brian Sykes' book, The $7 of Eve, in 2002. But at the same time as that was happening, two other things were happening. They sequenced for the first time, a group in Germany led by Svante Pääbo sequenced the first Neanderthal DNA. Ancient DNA was beginning in infancy in the early 2000s. And people were like, wow, we don't have to rely completely in living people's DNA. We can actually get DNA from not just ancient Homo sapiens, but ancient Homo neanderthalensis and other species as well. So in 2002, 2003, this group published the DNA sequence from the hypervariable region 2, HVS2 of Neanderthals. Really, that supported the idea that we were actually quite distantly related. And there was a push for many years. Are Homo sapiens a separate species from Neanderthals? Are we just a different subspecies? When DNA started coming in, with the DNA evidence in the 2000s and in the 2010s, the idea that they were separate species based on the difference in DNA started taking hold. And now most of us sort of argue that Homo sapiens is one species, Homo neanderthalensis is another species. Nonetheless, they could breed together. So it breaks down the biological species concept, which uses the ability to breed and produce fertile offspring as delineating what a species is. So again, all theories of what a species is begins to break down when we start doing ancient DNA. But what happened as well in the early 2000s was the Human Genome Project. And we sequenced the human genome, not just mitochondria anymore. People actually were looking at the entirety of our genome. By 2002, 2003, we now had a couple different genomes published. People started saying, well, we need to be looking beyond mitochondria and Y chromosome and looking at the entire genome. So I put this sort of scale of what was going on. So around the early 2000s, that's when Family Tree DNA, Oxford Ancestors in Britain, a Geographic project launched in 2005. But on the other parallel line, you had the Human Genome Project, which is the entire autosomal DNA and mitochondria and everything, and Svante Pääbo publishing the Neanderthal mitochondria first. And it wasn't until the late 2000s that we get the full mitochondrial genome. Well, in the late 2000s, when you start getting other companies coming in as well, 23andMe and Ancestry by 2007, 2008, and they actually immediately were not focusing on mitochondria. They focused on the autosomal DNA. Family Tree DNA, I think, followed with that with the autosomal, as did National Geographic later on as well. Not National Geographic did not join into the autosomal work until about 2011, much later. But in the meantime, science had moved more towards autosomal, thanks to the work of Svante Pääbo, and I put there David Reich as well. Up in Harvard, now with ancient DNA, he's getting consistently great data, not from just humans. His group and Svante's group has done Neanderthals and Denisovans and other species. So it seemed like the field evolved in parallel. And by the late 2000s and early 2010s, it was autosomal DNA that started taking precedence in academia. I was even told by the time I got my PhD in 2010, they were like, you were basing it all on mitochondrial DNA. You're not going to be able to publish in anything big. You may have to aim lower. So that was, heartfelt for me because I really loved working with mitochondrial DNA. It was fascinating to me. I really understood it. It worked well. It was an easy story to tell, but yet a complete global story. But all this other company and some of the top researchers had gone towards the autosomal DNA route. So really, by 2010s, early 2010s, Genographic, which I was already associated with, Tree DNA, and other groups began to work much more closely in autosomal DNA, but never really resting, never giving up on the mitochondria and the Y chromosome work. I remember when I was at National Geographic just a few years ago, a TV program called The Geniuses, and they asked me, how can we tie geniuses to DNA? And really without being able to sort of sequence the DNA of we began with Einstein, and then we did Picasso and other geniuses. This was a TV show in National Geographic. We actually used mitochondria to match known haplogroups of different geniuses or famous people of the past to people living today. And that became a neat feature in the mid-2010s, late 2010s at the Genographic project. So people would find out how far away their mitochondria or their Y chromosome was related to say, Darwin, for instance, or to Lincoln, based on DNA that we had obtained from relatives of those people. But by 2016, my friend Roberta Estes, you'll hear from shortly, wrote something interesting on her blog, which I happen to find heartbreaking, but in a sense, let's not forget about mitochondria. She wrote Mitochondria, the Forgotten Test. And Roberta, I took that from your blog. I hadn't told you yet, but I found that sort of hit right exactly what I wanted to say in this point of the slide, that other scholars were telling me, why do you still work in mitochondrial DNA? Why is this so special? Why do we still do this? When now you can do next generation sequencing, we're doing autosomal DNA. And honestly, I don't see any reason to not do mitochondrial DNA genealogy, but also for research. It really tells an important story, and really, there's so much data out there. And some of the data needs to be corrected. Some of the data needs to be revisited from 20 years ago. From the 80s and 90s, people were doing mitochondrial DNA. We now can go back to those samples and redo them and really help fix situations that really didn't make much sense early on. So can we make the 2020s a comeback of mitochondrial DNA? And that'll be my sort of takeaway at the end when we talk about the Million Mito Project. I wanted to give a few examples of where mitochondrial DNA has been in research, academic research. So here's a couple examples in the Middle East. We always fascinated by this question of out of Africa, and by the mid-2000s, there were papers being published. And can we find the right L3s? Can we find the first M, the oldest M and N right there in Yemen? So here's an example, again, sort of using haplogroup frequency to answer that question in 2006. By 2010, we had actually brought in a lot more samples from across the different parts of the Middle East, Southeastern Europe, and into South Asia, and in trying to find the elusive L outside of Africa, or maybe a branch something between L and M and N to tell us a little more. But those questions really at the core of us human. When did we leave Africa? When did our species become a global species and not just a continental species? Those are questions that remain today in 2010. A colleague of mine here published in 2018 about haplogroup HV. When I began studying this, there was H and there was V, and now there's a new haplogroup called HV. Well, it's been around actually for about 15 years now, but he did a study across different countries. You can barely see it here. I'm sorry. 2018 published this across different parts of West Eurasia, and the Middle East, and the Caucasus, Turkey, to understand the origins of this macro group, one of the old one actually evolving shortly after out of Africa. So this is still relevant. We're still publishing. In Southeast Asia, this is relevant. This is why I did my PhD work. And in 2007, they were using networks, like I showed earlier, to understand the evolution between haplogroup M and haplogroup E. Now, most of you who have heard of haplogroup E in Y chromosome. It's actually quite common throughout Africa, but also throughout the Mediterranean, the male Y DNA haplogroup E. But I bet almost nobody has heard of haplogroup E in mitochondria. It's the fifth letter of the alphabet, right after A, B, C, D in the Americas. Those were used up by Taroni and others by 1990. E is not used till much, much later. Actually occurs in a few pockets of Indonesia and the Philippines. And this is one of the first papers to publish. So even a whole haplogroup really didn't have much recognition until the late 2000s. This is a group, Hill and al. Published the phylogeny of groups E1 and E2 in different parts of Indonesia and the Philippines. And this chart published by Suarez et al. In 2009, really we used a new concept, do sort of heat maps, understand where an origin is by frequencies of that haplogroup in a certain place. So not just networks, but creating heat maps that show sort of a center of gravity, a center of origin, really. He also was one of the people who was doing some molecular clockwork and calculating the estimate. So another person who held on to mitochondrial DNA as a good thing to study, even as we transition from the 2000s to the 2010s. 2013 is a paper I published trying to find the origins of people in Guam and the islands of Micronesia, and mainly Guam and the Marianas Islands. And I used the work done by Hill and Suarez before that to really place the origins of the group called the Chamorros of Guam. And because there happened to be two very rare branches of E, rare everywhere else in the world. Mind you, in Guam, these are 90% of the population. So if you are from that part of the world, especially the Marianas or Western Micronesia, E is the most common haplogroup, but elsewhere it doesn't really exist. I still find this very relevant. It's very interesting and very useful in academic research. I wanted to show you here a couple articles in the Caribbean where this is being used, mitochondrial DNA. So the 2012 was one that I worked on, looking at specifically two Native American groups in the Caribbean, haplogroup A and haplogroup C. And we used networks to understand sort of the origin, the age estimate, and the distribution across, this is in Puerto Rico primarily, and we could estimate ages of when they got to the Caribbean by using phylogenetic methods, including time to most common recent ancestor. In 2012, this was published, accepted. It was received well. By 2020, I was now working with a much larger group. This was published in Nature late last year, where again, we visited Puerto Rico, looked at both modern and ancient DNA, not just Puerto Rico, but across different islands in the Caribbean. And you could see actually, autosomal DNA is 99% of the story. It's 99% of our genome, but this was done primarily in Harvard by the Reich Lab. I collaborated with them and we published this by December of 2020. And you could see how mitochondrial DNA makes it into the summary at the very end of the description of this article, where when they looked at mitochondrial DNA, it sort of supported some of the autosomal work. So some people do not see the importance of mitochondrial DNA or less so, but even papers published as recently as 4 or 5 months ago in Nature are acknowledging the mitochondrial DNA story has a relevant part to play in the story. I want a couple more slides here before I send it back to Paul. I'm actually been involved with an academic group and have brought them over to the genetic genealogy group to explore the origins of haplogroup W. Friends of mine that do a lot of work in mitochondrial DNA in the Middle East, the Caucasus area, are very fascinated by this group because it shows high concentration in different pockets. One near sort of Pakistan, India, Afghanistan area, the Caucasus, Turkey have a separate pocket. And then Southeastern Europe, and then in Finland, it's very, very high. This is a complete strange distribution of this haplogroup, but it exists in high concentrations in several disparate parts of the world. Academics approached me, and I work kind of in this in-between academia. I work with them, and I work also the genetic genealogy community closely. We've joined a partnership here. We're doing research around this. Joran Rumsfeld, part of our mitochondrial Million Mito team, has helped me sort of work on this project as well. So there's many opportunities to really bring academics and genetic genealogy together. And this is just one example that we're just getting started now in 2021, exploring the origin by combining some of the research done academics and some of the access that we have now to full mitochondrial genomes, especially through family tree DNA, who doesn't. A couple of last things I want to touch upon. When we study mitochondrial DNA as opposed to Y chromosome DNA, we can actually study anthropological concept of matrilocality very well. So when you study a part of a molecule that is inherited strictly maternally, you can immediately answer questions about whether it's the women who move to marry outside of the group, matrilocal, or they stay matrilocal and the men move. So Y chromosome diversity would suggest that you have that the men are moving and when you have Y chromosome concentrations in one place different than another place, you would suggest that the men are staying and the women are the ones moving. And I put an example here of Melanesia. So this is part of where I worked, my dissertation work in the islands of Vanuatu and Papua New Guinea. And when you look at the mitochondrial DNA diversity, you could see that it's spread quite wide across thousands of miles from the islands of Samoa, Tonga, Fiji, all the way to Papua New Guinea. You see haplogroup B well spread, but even haplogroup P and Q, these Melanesian haplogroups are showing up all the way in the Pacific. Because women move a lot, especially in this part of the world. Melanesia is traditionally a very patrilocal, big men society where it's very patriarchal as well, where inheritance of power is passed on strictly through the paternal line. Men own land. In some cases, women cannot own land. It's very male-centered societies across this area of Melanesia. And you could see the movement of women quite clear when you see the spread of mitochondrial DNA haplogroups. However, when you look at the Y chromosome on the left there, you do see some more localized, maybe you see some dark purple, which represents, I believe, haplogroup O, whereas haplogroups M and S, other Melanesian groups, seem to be quite local to different parts of Melanesia. So you do see some movement of Y chromosome, but it seems to be slightly more localized. So there is a connection there between understanding the pattern of paternally inherited Y chromosome, maternally inherited mitochondria, and these cultural practice of patrilocality, of patriarchy, and so forth. So Melanesia is a fascinating place in the world to do this kind of work. And that's actually still done a lot by researchers from Australia, from Indonesia, as well as from all over the world. Another thing that really is good to study together with mitochondrial DNA is languages. So I put here a chart my colleague Dr. Tad Shur used in when studying mitochondrial DNA haplogroups across different linguistic groups in the Americas, mainly Native American groups. And he claimed to see sort of a strong pattern between the frequency of some haplogroups and the major linguistic groups. For instance, the Eskimo Aleut groups seem to be primarily an A and a D, a specific branch of A and D. The Nadine are primarily this group A, as you can see. These are the Athabascan groups in Alaska, Canada, and a small pocket in the southwest of the U.S. On contrast, the Amerindian group, the largest group, very diverse group, possibly the oldest group of linguistic group in the Americas, carries all the major haplogroups, A, B, C, D, and as well as X. So there is some connection, he argued, between mitochondrial DNA and languages there. Across Eurasia, this is done as well to understand, and this is an example from Y chromosome. Again, I could not find the best one, but when you're looking at this parental data and language groups, people have used it to understand the movement of R1b from Asia to Europe and the association with the pre-Indo-European, PIE there languages, or R1a possibly associated with Uralic languages and haplogroup N, Y chromosome haplogroup. So this is very useful. We can actually make trees, phylogenies of languages, and then we can make phylogenies of mitochondrial DNA and Y chromosome and put them together, see how well they line up, see how well they map. And this is where anthropology is right now with a lot of this work. Then the question, though, comes back. If you see the pattern with mitochondria, is it because the mothers are the ones teaching the children languages? You would imagine the mother is the one teaching a child to speak, and should you speak your mother tongue, therefore mitochondria should be most closely tied to languages. Whereas we see patterns, clear patterns right here in the Y chromosome as well. So it's not clear. Again, this gets me excited about anthropology and genetics. If nothing is so clear, that really asks and begs questions when things don't necessarily follow a pattern, yet they're close enough that you can make strong arguments in one way or another. So anyway, I think this is all I have right now when it comes to sort of mapping, anthropology, and genetics. So I'm going to pause here and give it back to Paul.
All right. Thank you, Miguel. That was awesome.
You're welcome.
Yeah, thank you. I love listening to Miguel talk about frequency distributions of haplogroups. I could listen to it all day long. It's really fascinating to watch these groups travel across the landscape and sort of make up stories about what happened and how linguistics are coupled to the haplogroups or coupled to populations, or if it's patrilocal, matrilocal. And I really liked what he said about archaic humans and sort of the history of how we thought about that. So I'm going to just kind of cap that off and kind of throw a bone to the ragtag team of Mito misfits here and show one example of how mitochondrial data gave us a story that we wouldn't have with nuclear data alone. So to do that, let me just kind of set up what we know about ancient and archaic DNA. This is kind of as of a few years ago. I think this is a review paper from 2017. It shows Neanderthals and Denisovans. Like Miguel said, the whole genome sequences were published in 2010 by Reich's lab, David Reich and his collaborator Green. We knew from about a decade ago that there was this kind of ancient branching pattern of, at least nuclear pattern of archaic humans splitting off from human beings, modern humans, and then Neanderthals splitting off from Denisovans. And an old standing question was, are we all sort of evolving into these lineages at the same time, or is there one origin of modern humans that then does or does not interbreed with archaic forms of humans, such as Neanderthals? And so initially, when we looked at the mitochondrial pattern, we found, okay, well, there's no Neanderthal or Denisovan haplotypes in human beings, or at least modern human beings. So it looks like they stayed separate. But then, of course, when we started looking more closely at the nuclear pattern, we found this 1 to 2% of Neanderthal and Denisovan ancestry in humans across Eurasia. So it's been sort of from one pattern assumed to another pattern, and now it seems like it's nuanced. It's somewhere in the middle, that there was an ancient split and then subsequent minor gene flow. And then Michael Hammer, who's one of the founders of Family Tree DNA, and who's a collaborator of ours, kind of added this other additional super archaic gene flow event from probably within West Africa. So, the story grows by the year. So for Neanderthals, what's really interesting is, yes, we've kept some of their ancestry. We've kept some of the important gene regions. So if you look at the actual genetic material that we retain, it tends to be genes that are important for things like skin color and hair, keratin, immunity genes, which may have been important in the early days of the last glacial maximum. And then in Denisovan admixture with people in Tibet, there's this adaptation for high elevation as well. And so you find these blocks of nuclear DNA, they're kind of islands of nuclear DNA, but for the most part, we've kind of purged everything else. So Miguel mentioned the biological species concept. That was an idea from Ernst Maier in 1942, I think, where he basically said if two species are separate, then there's zero gene flow. And of course, this is evidence that we've had some gene flow. But with, a more modern look at things, we could say, well, there's gene flow, but there's not a lot, because most of the admixture between Neanderthals and modern human beings has been purged. So what you see is on the left here, the sort of unimportance of the gene correlates the amount of Neanderthal ancestry that we have. So the more important it is, the less Neanderthal admixture we have. And this is particularly true for certain places in the genome. It's true on the X chromosome, which tends to adapt faster. Natural selection acts more strongly because there are more conflicts between the X and other places in the genome. So it's 5 times stronger there. Also for the FOXP2 gene, which we think is very related to human speech, is the least amount. It's very absent of Neanderthal admixture. So clearly there's this sort of admixture pattern around 40,000, 50,000 years ago, and then slowly we've been purging a lot of that ancestry. And you can see this with ancient DNA as well, except for this one little blip here. This is the OASE-1 sample in Eastern Europe. This is a recent hybrid. So I think this is a human being who had a great-grandparent who was a Neanderthal. So 12.5% Neanderthal roughly. But for everyone else, through time, you can see that there's this gradual decline in Neanderthal ancestry, supporting that pattern that there's lowered fitness for much of the DNA that they contributed. Not all, but a lot of it. And even simulations have kind of shown that the frequency of mating between the two species was very seldom. Only about 1 to 3% of our genome has any Neanderthal DNA. And by simulations, it seems like we may have mated with them in Western Europe anyway, every generation or so, maybe 1 or 2 generations. And the pattern bears that out. So in much of Eurasia, we have about 2%, ranges from about 1 to 4. Because of the locations of the multiple Denisovan interbreeding or admixture events, it seems like there may have been 3 total, depending on the population you're talking about. It maxes out at about 5% Denisovan in Papua New Guinea, in that area, the Sahul subcontinent. But most people in Asia have much less, less than 1%, about 0.5% or so. And then I mentioned Michael Hammer, one of our collaborators. He released a paper some years ago showing that there was this archaic or super archaic African introgression event that may have come from West Africa. This is particularly interesting just because it may have to do with our current Y atom. Our Y atom is about 250,000 years ago removed from the rest of the tree, whereas the Y atom previous to that, A0T, was only 150,000 years removed, which is, corresponds better with mitochondrial Eve, who we also think is about 150,000 years removed. So that's an interesting topic for further study. So here's the really interesting thing we learned from mitochondrial DNA. So back in 2016 and 2017, a couple of research groups showed that there was this admixture event, not from Denisovans or Neanderthals into modern humans, but rather from pre-modern humans, the humans that led up to us, but were 380,000 years ago. From those stem, you could say, modern humans into Neanderthals. So this would have been from people in Africa before modern humans into Neanderthals in Europe. And this is inferred because there's this sequence called the Cima de los Huesos sequence in Europe that is most closely related to Denisovan sequences, mitochondrially speaking. Nuclearly, though, it's like Neanderthal sequences. And the rest of the Neanderthal mitochondrial sequences are more closely related to humans, modern humans, than they are to Denisovans. So here's an example. We talked about incomplete lineage sorting and drift a while ago, right? So here's an example where the nuclear pattern shows humans are the sister group to Neanderthals plus Denisovans. But mitochondrial DNA shows the opposite. It shows that humans and Neanderthals together are the sister group to Denisovans. And this sample, this one Cima de los Huesos sample kind of resolves that because it shows that although nuclearly it was Neanderthal, mitochondrially it was more like Denisovans. And so based on this, we can infer, and this is based on our own work, we can infer that the timing of this was around 380,000 years ago to about 280,000 years ago. At some point, there was this gene flow event from pre-modern humans into Neanderthals, at which point all Neanderthals after that had a more human-like mitochondrial sequence. So this is really cool, and you would need both nuclear and mitochondrial data to figure this out. All right, so I'm going to cap that. I'll give a brief overview of heteroplasmies, and then I'll give it away to Roberta, who's going to talk about all the genealogical cool stuff. So there are, like I said before, a couple of types of heteroplasmy. We know that there are multiple mtDNA populations, between cells, within cells, and then there are multiple mitogenomes within a single mitochondrion as well. So due to that high mutation rate we talked about, often heteroplasmies are undetectable. They're at a very low rate of less than 1%. So it's actually fairly common. The majority of people have very, very low heteroplasmy levels, often below the level of detection. It's only when you get to that, 50, 60, 70% or more level that you can start to have problems. And there can be SNP heteroplasmies as well as indel, or we call them length heteroplasmies. And there are kind of two ways that the human body gets rid of heteroplasmies. One has to do with when cells divide. There's a drift event that reduces the heteroplasmy and reduces its frequency in the next daughter cell. And then there's within a single cell, when mitochondria divide, the same thing happens. They sort of have a drift event, only some of the mitogenomes get passed on, and there's a different frequency in the next daughter mitochondrial organelle. So here's a really interesting result in the last couple of years. This is from Way 2019. They showed that new haplogroup heteroplasmies match nuclear DNA more than mitochondrial DNA. So in other words, when there's a new heteroplasmy, and every new mutation has to start as a heteroplasmy at some level, it's more likely to resolve based on what the nuclear DNA looks like. If the two mitochondrial and nuclear DNA have been together, have been matching for a long period of time, the heteroplasmy is likely to resolve in a way that's compatible with the nuclear background. And that's fascinating because that could really have an influence over the branching pattern of the tree over time. And so known variants are basically transmitted more than novel variants. So there's a bias there. There's another paper that shows that there may be selection against heteroplasmy causing this uniparental inheritance, this matrilineal inheritance that's characteristic of the whole molecule. So, malformation or lowered fitness due to heteroplasmies in general may be the reason why we have this inheritance pattern in the first place. Usually these things are unstable and they resolve within a few generations. Usually a few means 1 to 3. But occasionally we have these interesting examples where they seem to be stable. So this is from some Swedish genealogical research. Peter Sjöland did some research on this family going back to the 1600s in Sweden. And there are 130 relationships that are confirmed. They're all in haplogroup H52. And so there's this heteroplasmy that seems to be inherited from one generation to the next generation consistently and has not resolved in that time. So there's a lot that's still not known about how long they can persist. The hotspots for heteroplasmies tend to be in the control region. So in HVR1, in positions from 16093 to 16337, HVR2 from 72 to 279, and length or indel polymorphisms, heteroplasmies tend to be at the sort of poly-C regions. So we have a poly-C, meaning multiple Cs in a row, at 16189, and the region from 309 to 315. Like I said, ultimately all novel mutations begin as these germ cell heteroplasmies, and they either resolve one way or the other, and nuclear DNA may play a role in how that happens. As far as genealogy, don't discard them. Don't think that they're not important. They may be novel mutations in the making, and their shared pattern with distant or close relatives may tell you a lot about how the tree is structured. So that's my bit. And with that, I will turn it over to Roberta.
Hi, everybody. So what I'm going to do is I'm going to bring this back together. And I want to take a minute and do a couple of things. First of all, as one of the Million Mito team members, it's absolutely awesome and amazing to get to work with Paul and Miguel and Joran, who's been deathly silent during this so far today. But This is so exciting and so interesting, and there's so much to learn. It's just been a journey of a lifetime. So I just want to say that and to thank both of them for their efforts. Somebody, I don't know if it's Paul or Miguel, said two things that I thought were really interesting. First of all, that we're connecting the dots of history, of human history, and we are. And the great thing about mitochondrial DNA is it can connect us genealogically, but it can also connect us back in time in terms of our ancient history. And it can tell us so much more than autosomal DNA can when we're beyond just the first few generations. Second of all, I had to laugh. Somebody said, it would seem simple enough. Well, you know what? It would, but it's not. So what I'm going to do is I'm going to talk to you about 3 or 4 ways, a few case studies, how I've used mitochondrial DNA for genealogy quite successfully. And this is my mitochondrial lineage because it's just me and I'm geeky that way. I will say that at my blog, there's a bunch of articles. There's more than 1,400. It's all free. And there's a special page set up for mitochondrial DNA. So for genealogy, first of all, every father has a mother. And I know we all think in terms of ourselves and testing our own mitochondrial DNA, but that's not always the case. And we don't have to just test ourselves. You know, we're genealogists and we have other people in our family, we have cousins, and no matter how you look at it, half of everybody's genealogy is maternal, or actually matrilineal, because it goes directly back to the mother's side. And that reflects your mother's half of the tree. And every male ancestor had a mother. So one of the things that I'm really careful about when I talk about mitochondrial DNA is the word maternal and matrilineal. Because people say, oh, you know, I had a Native American ancestor on my maternal side. Well, they may mean anything to the right half of this black dividing bar here. That's maternal, it's on your mother's side, but that's vastly different than matrilineal. Matrilineal is the direct mother to mother to mother line. So I'm always careful when people say something, make sure that we're talking the same language. And that's also an issue sometimes when people enter their earliest known ancestor. It's, you know, it says at Family Tree DNA, it's like on the paternal side or on the maternal side. Well, you know what? On the maternal side, if your earliest known ancestor is your maternal great grandfather, you could put that in, but that's certainly not relevant to mitochondrial DNA. So the test relevance, obviously to obtain your mother's side information, your matrilineal, the Y-DNA isn't useful. Autosomal divides in every generation, so that's reduced every generation to only a fraction of the ancestors, but mitochondrial DNA never divides. It's not diluted across time, and because it does have occasional mutations, that makes it genealogically useful. We have a direct line of descent. We know who it comes from. We're not testing autosomal DNA. And you know, I do love that. I write about it all the time. So this isn't a negative about autosomal. These are just 2 different kinds of DNA, 2 different kinds of genetic tools for 2 different circumstances. And the great thing about mitochondrial DNA is we don't have to guess who it comes from. We know who it comes from. It comes from that person, the tester's direct mother to mother to mother line. It's inherited by both sexes of children from their mother. And in the current generation, people can test males. You would not believe how many people don't realize this. I see this all the time on Facebook. It drives me insane. They say, oh, no, it's only for females. No, you inherit it from females. It's only passed down by females. But in the current generation, males can test. You get your mothers. So the mother always contributes the mitochondrial DNA, except for the very rare occasion when we do have father inherited, and those people have very severe medical conditions and generally don't survive very long. So that's not the case. If you hear someone say that, it's not the case. It's not mixed with the DNA of the father like autosomal. It's passed intact, and every genealogist can test their own mitochondrial DNA. And I see people say, well, don't bother with that. I only got a few matches. Well, first of all, you don't want thousands of matches for mitochondrial. You want a few really good, relevant matches. And it is sometimes a waiting game. But if you would think about it, If all the 20 million people who have tested their autosomal would test their doggone mitochondrial DNA, people wouldn't be saying it's not relevant, because look at all the matches you'd have. And if they uploaded the trees, you'd be able to figure out how you connect. So it's kind of that self-fulfilling prophecy. So for genealogy, we want to answer the question, where did I come from? That's what everyone really wants to answer. Where did I come from? Who are my ancestors and where were they? So let's look at unique mitochondrial DNA information in our case studies. First of all, haplogroups. Second of all, matches. Okay, locations beyond surnames, and trees finding connections. So for haplogroups, matrilineal clans identified by specific DNA mutations and locations. And I think Paul and Miguel covered this. We can tell based on the haplogroup what world region the person came from. At 23andMe and Living DNA, today you can obtain a base haplogroup from those companies. And Family Tree DNA has announced this for Family Finder customers sometime in the future. But that's an estimate, and it's an estimate versus a detailed sequence that you can get from a full sequence test at Family Tree DNA. And how do they obtain that estimate? Well, they were talking about probes earlier. What they did is they said, okay, we're going to take 1,000 locations on our chip of 700,000, and we're going to test for those specific locations. And those are haplogroup defining locations. So that's what they're doing. They're not testing the whole mitochondria. They're just testing specific locations that give you a base haplogroup. There's nothing wrong with what they're giving you, so long as they're testing for all the right positive and negative locations. So for me, the difference was I obtained haplogroup J1C at those companies, but my full haplogroup today at Family Tree DNA with full mitochondrial sequence is J1C2F. So no matter where you tested, you can look at the public mitochondrial tree at Family Tree DNA. If you go to the main page before you sign in, just go to their familytreedna.com page and scroll all the way to the very bottom. The mitochondrial tree is one of those links at the very, very bottom. And if you click on that, it takes you to the haplogroup L, which is Eve, mitochondrial Eves. And you type the branch name. So for me, I typed in J1C2F, but even if I had J1C, you can see over here where I could have just typed that in. And then it shows you all the countries where J1C2F is found in the Family Tree DNA database. And you can see here that it shows you that there are 11 plus 11 here. So you could click on this link and you can get more information. For example, are the locations where J1C2F, whether I match them or not. Now remember, this isn't just my matches, this is anybody in J1C2F where that haplogroup is found. So it's found primarily in Sweden and Norway. You can see that here. My ancestor was found in Germany. There's 5 people. And then there's a scattering of other locations that you can see. So this is a piece that you can use in trying to piece your tree together, whether you've tested your full sequence or not. It's just a matter of how far up the tree the haplogroup is that you're researching. So let's talk a little bit about this, how people make haplogroup discoveries that are relevant for genealogy. The first one was when Anne Marie was identified as a Native American ancestor. So the original tester is an Acadian descendant. The Acadians were settled in Nova Scotia in the 1600s and intermarried with the native people there, the tribal people. And Anne Marie was born in 1631 and married Rene Rimbaud. And this person who actually took the original test did not know that Anne Marie was Native American, but she did. She took a test and she discovered that Anne Marie was A2F1A and that she's Native American. Well, the really cool thing is I happen to know this person, but I didn't know that Anne Marie was also my ancestor at the time because I had not broken through that brick wall. So when I broke through that brick wall and I discovered that Anne Marie was my ancestor, I also discovered that the tester who I actually knew was my cousin. So is that not cool or what? You know, that's like using the haplogroup to define where the ancestor came from. We also now know why Anne Marie didn't have a surname in the Catholic records. The person had to convert to Catholicism before the priest would marry them. But she had no surname because she was Native American. So they just wrote her in as Anne Marie. So I looked in the Acadian Amerindian Project. I'm one of the administrators. And so I looked at the matches for where else was A2F1A found. And this is where other places where people who matched and were of that haplogroup were found in the project. So we can see there are several in Nova Scotia and Maine, and then on to the west. And some of the work that we're doing with the Million Mito Project will expand on this. So let's look at Native American haplogroups, because so many people, they want to find their Native American roots. And mitochondrial NY DNA testing is one way to do that. You can test for autosomal all day long, and you may or may not show anything, but no matter how far back in time that Native American ancestor lived, if they exist, their Y and mitochondrial DNA haplogroups are going to be Native American. They're not going to reduce in time. They're not going to be washed out or divided. So they are subsets of A, B, C, D, and X. And what I do when people say they have a Native American ancestor, I ask them to find someone in the correct line that they think that person descends from. And this is actually one of the reasons why I originally tested myself and specific other cousins, because the family oral history was that my great grandmother on my father's side was Native American. And so I found someone to test her mitochondrial DNA, and she's not Native American, at least not from that line. So I have consistently found someone from every single ancestral line that I can find, and I'm still missing a few, and I will, because you're always at a brick wall at some place, to test for every one of my lineages. And I have confirmed several Native American ancestors that way. So this is one of the reasons that you need more than a general haplogroup, because for Native American people, A, B, C, D, and X, and also for the males and Y, Q and C, there's only subgroups of those haplogroups because those haplogroups were born in Asia. So only certain subgroups came here. So you need more than just a base haplogroup to determine if they're Native American. And also you want to be able to look where those other people are from that you're matching, because that's relevant to your genealogy. So let's take a look at haplogroup matching and maps. This is one of the people that I work with who's Native American, and they are haplogroup B. And she was trying to figure out what tribe she might be from. And we say over and over again, you can't tell a tribe, certainly not from autosomal. But you know what you can do? You can figure out where people are from. And if you know the history of the area, you can figure out more than that. So this person was haplogroup B, a subset of haplogroup B. And look here, all of her matches, and these are full sequence mitochondrial DNA matches, exact matches. Look where they cluster. They cluster right here in Colorado and New Mexico. And there is that person. She's the white pin behind these red pins. And if you look at all the people that she matches in this cluster, they're Diné, they're Pueblo speakers, and they have a very distinct tribal lineage. And so will she ever be able to join a tribe? Well, no, not from this, certainly. But can she learn more about her history, the history of her ancestors, the history of where they lived and what they were doing and where they came from and where they migrated from? She most certainly can. So from her own personal matches, now she needs to go and look at that public tree to see where the migration pattern of this haplogroup was, where it came from, and when it was in those places. So this is really very exciting, even just from something like a map. So how did she get those map locations? Well, this is a plug for every single person here to go in and enter their map locations. Because if it wasn't for those map locations that showed her where those other people, where their earliest known ancestors on the matrilineal line were from, she wouldn't have been able to garner that information. So if you go in to your account settings here, Family Tree DNA up in the right-hand corner, and there's account settings, and select genealogy and earliest known ancestors, and then enter your direct patrilineal and your direct matrilineal ancestors. And you don't have to figure out the longitude and latitude, just click on the update location here, and it takes you to this map where it steps you through. If you click on edit locations, it steps you through the names of those ancestors, and you type in their location like Deal, England, or wherever, and it puts in the longitude and latitude, and it backfeeds this. So please do that, because if all 273 of my matches here had done that, then it would be much more useful information for me, and it would be for everybody. So I do this. Mine are obviously in there because this is mine that I'm showing you. But I also email my matches from time to time, and I send them information about this. And I say, look, if this information is useful to you, and if you would add yours, and I can tell that they haven't because the earliest known ancestor field's not filled in and they're not on my matches map, then if you will add yours, then it will help us all. And you know what? It's amazing. They do. I don't just ask them to. I explain to how it will help them and how it helps everybody. And then they're much more inclined to do that, whether they answer me or not. Okay, so let's move on. So let's talk about mitochondrial DNA matches and what you can do with this. First of all, to be really relevant, people really need to take the full sequence mitochondrial DNA test. This is an autosomal DNA, and so they have to understand there's a difference, and not everybody does, and they don't understand what that difference means. I tend to ignore, and I should say at my own peril, HBR1 and 2 matches. Those tests aren't offered anymore, and thankfully, because they're not terribly relevant, because there's a lot more mutations in that HBR1 and 2, and only full matches, meaning no mutations, are counted. I should sometimes go look and see if someone's added a tree and what their earliest known ancestor is, but I tend to be lazy and only focus on the full sequence. I actually occasionally encourage one of them to upgrade depending on where their ancestor's from. So you match with other testers, and this is my HBR1, 2 encoding region, so the full sequence, everything. And I always look to see who has provided the trees, and I'm really excited because look here at all these tree icons and the earliest known ancestors. And the exact matches in this case start with a GD, or genetic distance, of 0. Well, what does that mean? The genetic distance of 0 means that they're an exact match. In other words, for me, and I'm showing you my sequence here at the right, the haplogroup defining sequence for J1C2F, and I'm only showing from J to J1C2F. Remember, J is a subset of an earlier haplogroup, and so there's a lot of accumulated mutations between mitochondrial Eve and J, and I'm not showing you all those in this. I'm only showing from J to J1C2F. So these are the mutations that define each one of these various haplogroups. So you can see the company that gave me J1C probably tested for some subset of these and 14798, but didn't test for 188 or 9055. So, and that's okay. It's not a criticism. It's just education. So we know that haplogroup J1C2F is about 1926 years old with a standard deviation of 3,000 years. We know that from Duran Bahar's Copernican paper in 2012. So what this means is, on the average, it's probably about 2,000 years ago that mutation at location 9055 occurred. But a standard deviation of 3,000 years means it could be 3,000 years further back, or it could be even closer in time. So I just say about 2,000. That is the best estimate that we have based on Bahar's paper in 2012. The Million Mito Project, of course, one of the goals is, after the tree is recalculated, to also recalculate aging. So all the other mutations that I have are listed under the RSRS on my page as extra or missing. So I've added those down here in yellow. The mutation at location 315.1, or any 315, is not used for matching because it's in an unstable region, but they're still shown. So what we have here is, this is my whole list of mutations. So a GD of 0 means that everybody that has that GD of 0 matches me on every single one of those locations. And they don't have any mutations I don't have, and I don't have any mutations they don't have. We're an exact match. So those are interesting because I want to look at where their ancestors were from and what time frame to see if I can find out more information about my ancestors. So we have things called heteroplasmies, and the heteroplasmy, as you know from Paul's talk, is where you have two different values, like a G and an A or a C and a T in the same location. And if you have a heteroplasmy, they're generally recent, but not always, and they may cause mismatching, or not, but not always, depending on how you match or don't match them. So those is information you may need as you go and move from your GD of 0 to your GD of 1 or 2 or 3 matches. So what does a genetic distance of 1 or 2 or 3 mean to you as a genealogist? A genetic distance of 1 means one mismatch, but it's not necessarily the same. So it's really easy to look at that list of people and see all those people with a GD of 1 and assume that those people would all match each other, but that's not the case. So what I did is, over the years, as I have new matches, I write to these people, because we can't see each other's mutations at Family Tree DNA. And at Mito DNA, you can see the HBR1 and 2, but you can't see the full sequence. So I have been writing over the years to people and asking them to share with me their mutations, usually just a screenshot, because that's the easiest thing to do, because otherwise it's like I just want the ones that are really extra and missing, because those are the ones that are going to be different from the haplogroup and from me, right? So when they do that, I constructed a spreadsheet where I group them together. So this is part of my spreadsheet. Obviously have their names, and I'm obviously not showing that for privacy reasons. But what we have here is, these are my first few matches as I group them on the spreadsheet. So these are all a genetic distance of 1. But as you can see, the first 2 people here, they have a mutation at 15574 where it changed from a C to a T, but I don't. But they would match each other, full genetic distance of 0, they would, because they share the same exact mutation that I don't share with them. So if you look down, the next 3 people share a mutation at 1719 where they differ from me. So that would be a little subgroup too. And then if you look down at the next group, this one's even more interesting, and it's a little more complex, because this is, the first 3 people are a GD of 1 because they mismatch me but match each other on the same 13434. But if you look at the next person, who's a GD of 2, they mismatch me on 2, and one of them is where C195 is missing in theirs, but not in mine. And then the next person is a GD of 3, where they share those first 2 mutations with me, or they're different from me. And then they have a third mutation that's even different yet. So what you kind of see here is maybe another mitochondrial DNA subclade, where 13434 would be the major subclade, and 2 little baby ones that are sprouting at the bottom. But the only way to find your mutation information is to actually contact your matches. And the way that I have found that you get the most response is if you contact them when they first get their results back, then they're much more interested than they would be a few years later, maybe when their emails expired or something else. So that's what I've done over the years, and it's actually been very successful. And I'll show you why in a minute. This is my matches map. Now remember, my earliest known ancestors found here in Germany. You can see the map at the bottom down here. And these are my full sequence matches, where red is an exact match, orange is one difference, a GD of 1, and yellow is a GD of 2. So these locations provide really important clues for me. But why are my exact matches in Scandinavia? when my ancestors in Germany in the 1600s. Now I know this is my ancestor. I have proven records, meaning birth and death and baptism records back to this ancestor. So this is not, I didn't go and, scrape somebody's tree and just attach this person. I absolutely know this for a fact. And guess what? The records disappear in the later 1600s, and the records disappear because the 30 Years' War came right through that area of Germany. So the churches were burned, and the population, parts of Germany were depopulated entirely. So there weren't any records before that. But maybe the 30 Years' War had something to do with that, you think? Well, guess what? Math is your friend. Because if haplogroup J1C2F was born about 2,000 years ago, and I have 4 mutations in the regions that happen about every 400 years because then they're in the hypervariable region, certainly something that happened about 1648 would be reasonable and could explain why I have so many full sequence matches in Scandinavia. And then when I look at those little mini groups that I showed you before, guess what I found? See this group up here in northern Sweden with these exact matches? Yeah, guess what? That's where some of those are that all match each other and form that little mini clade. So I think we might be onto something there. But isn't that fascinating? Because I hit the end of records in the 1600s, but thanks to actual historical records and my mitochondrial DNA exact matches, I can piece something together. Because one of the reasons I wanted to know if my ancestor came from the Scandinavian group or the Scandinavian group came from my ancestor, and piecing together my own little phylogenetic tree showed me unquestionably that my ancestor descended from the Scandinavian group. So that was critically important for my genealogy. Even though I'll never know their names, I know where they lived and where they came from. The fourth case study is climbing trees. So you may have people, first of all, that match you on both Family Finder and mitochondrial DNA. So the first thing I do is with the full sequence matches, I go in and I use the advanced matching and select Family Finder and the full sequence at Family Tree DNA, and I select people who match you on both, and I say yes, and I look to see. Now that doesn't mean that you match because of the same ancestor, but it's a hint. It's a place to start. So that's what I do first. You may have ancestors who match in trees with you or match in trees with each other. And Family Tree DNA doesn't have tree matching today, but Genetic Affairs, which is a third-party tool, combines mitochondrial Y and autosomal DNA and tree matching. So what I did, and this is not mine because I don't have one that's a good example, but this is somebody who said that I could use their example, and they are a mitochondrial DNA match here. And they're also, and here's their common ancestor, and they also had several autosomal DNA matches down here that I cut off. But this is an example of where tree matching can provide you with incredibly useful information. And I wrote about that in this article on my blog. So sometimes, because we're genealogists, we don't get the easy answers. Because if you've done all this, the easy answer is probably not evident. But I learned that if you build out other people's trees, you can break down brick walls. And that's really what we want to do in this. We might want to confirm what we already think, but what we really want to do is break down those brick walls. So my cousin asked me, and she was focused on this line, and she wrote me and she said, can you go back and look at our ancestor, Phoebe, who was born about 1750 and married Jotham Brown, but we didn't know what her maiden name was. Can you go back and see what you can find in her mitochondrial DNA test? Because she knew we had that for a descendant, because I'd found someone and paid for her to test, and we had it. And I'm like, oh, fine, I will. But there's nothing there. I already looked. Well, guess what? I was wrong. So I went back and I looked, and I didn't see anything, because what do you look for? I didn't know her maiden name. What am I going to look for? But what I did notice with something really interesting. Look here, the very first thing. See Lydia? Well, the reason I noticed this, first of all, it's a GD of 0. All right, so it's an exact match, even though even a GD of 1, if it's a heteroplasmy, you don't want to discount it necessarily. But Lydia, that name was interesting because my ancestor named her daughter Lydia. And Lydia isn't all that common a name. It's not like Elizabeth or Mary. It's different. And they provided a tree. So I went and looked at their tree. And this is all that they provided. They didn't provide the marriage information. They didn't provide parents. But then I went to Ancestry and My Heritage, and I looked up William Powell Simmons and Lydia Jane Cole. And I found information for both of them, including this tidbit right here, that they were married in 1801 in Frederick County, Virginia. Well, guess where my Phoebe and Jotham Brown lived? In Frederick County, Virginia. Well, that's kind of an interesting circumstance. I mean, is that a coincidence? I mean, it could be, but so then I started digging on Lydia. And I called my cousin and I said, okay, I don't know, but we might have something here. So then we went back and we found Lydia Cole, this same Lydia Cole, who was in Morristown, New Jersey, and her father was John Cole. And I'm like, oh God, it's not the same one, because New Jersey to Frederick County, Virginia, that's not a migration path. And then the cousin, who's a better genealogist than I am, said, whoa. I just tracked the Quaker Church members in Frederick County, Virginia, where we know that Phoebe and Jotham were members, and a whole bunch of them came from this church in Morristown, New Jersey, together. And then she found John Cole's children were all baptized in 1755, the first several, and there's a record that they married a Jotham Burt. Well, the original record is terribly, you can't read it. Is that Jotham Brown? Is this the same family? Well, it certainly could be, and now I'm super interested, right? So I went over to Ancestry, and on my through lines, on my tree, as a experiment, I added John Cole and Mary Mercy Kent as my Phoebe's parents as an experiment. And guess what? I immediately got 49 through line matches. Now, remember, through lines is a hint, but 49 hints of other people who match my DNA, who descend from other children of this couple, is a pretty doggone good hint. So I contacted those people because I wanted to see if these segments triangulate. And some of them were already at other vendors, at Family Tree DNA, My Heritage, or GEDmatch, where I could triangulate, and others weren't, but they were willing to transfer. And sure enough, these segments triangulate. So because of mitochondrial DNA and genealogy, and a cousin who was smarter than I was and looked at the migration pattern for the church, and then the work that I did through records and then attaching them on through lines and then triangulating them, I was able to break down that brick wall for Phoebe, whose last name was Cole, and I pushed it back several generations through her parents, who are fairly well documented. So that's an amazing success story. But if I had just given up, which I had done, and ignored those mitochondrial DNA matches who didn't have their trees built out, I would never have found that, because I didn't know her last name. So I had to do the work and then do the segment work to figure out if that was actually the right line. And it was. So that was an amazing breakthrough. But it took work. It wasn't a freebie. It wasn't a gimme, but it was there. So what's the message here? Check or test all your mitochondrial DNA lines. You don't know what you don't know. You can discover Native American, Jewish, African. You don't know. You literally do not know what you don't know. And target testing provides answers. And so that's what I do. I find people who descend from these ancestors appropriately to test their mitochondrial DNA, and I offer scholarships to people to test. They don't all take me up on it, but if they've tested at 23andMe or living DNA already, they already have at least a base haplogroup, so you can tell something. So where do I find these people? Well, I target my first 8 lines, my big 8 here, and I go to WikiTree. WikiTree has a great service where you can enter that you are mitochondrially descended or Y descended from any ancestor, or that you've autosomally tested. And I've done that for all my ancestors, but I go check all mine where I directly descend mitochondrially. But I go check those other ancestors to see if anybody else has entered that information, because that's really important. And then you know who they are, know what their mitochondrial DNA is. You can also find people that haven't mitochondrial DNA tested there and ask them if they're willing to. I use through lines like I just showed you. I use my heritage theory of family relativity. Anyone who's descended from that couple through all females to the current generation, where males can also test, is a candidate. So that directs my research. And so this is just an example of what you could find. These are not my results, but you could find all different kinds of information that informs you about where to search and the history of those ancestors, even if you don't know their names, even if you don't know their surname. So here's some resources. At DNA Explain, I created a page you can see over here on the right. It's a mitochondrial DNA resource page. You can subscribe to the blog for free, but even if you don't, if you go to that page, it's a compendium of all the articles that I've written about how to use mitochondrial DNA and that kind of information, and it's there for you. And also know, I think you can search the blog. So that's it for this part of the presentation. I think what we were going to do next was talk about the Million Mito Project. And I put together a little base slide group for that. So if you bear with me a minute, I will bring up that presentation. So first of all, I want to give Paul a really big thank you, because, you know, I really apologize, but you've probably already guessed that we're really geeky about this, and we all love mitochondrial DNA. We really, truly do. I remember the day that I met Miguel, and we were talking. It was at the conference at Family Tree DNA. I was just standing there and I'm like, I just love mitochondrial DNA. And he looks at me and goes, so do I. And we like bonded in that minute, because I think we might have been the only 2 people in the room, you know, who really, really loved it. But then Paul joined in, and now there's 3 of us, and Joran too. And, you know, getting the 4 of us in the same room, it's scary, scary stuff. But what the throw out I want to do here, the thank you, is that Paul created this image of the mitochondrial DNA and the powerful women, because the idea or the goal of the Million Mito Project is to rewrite the tree of womankind, to trace it and to tell us more about it. So thank you, Paul, for that really cool graphic. I love it. So I'm going to talk about this, and I want to invite Paul and Miguel if they have something to say, because it's hard to do. This is all of our story, and Joran too. This is all of our story, the 4 of us that are colleagues. And it's hard to do one presentation with 4 presenters, especially in a circumstance like this. So these are the 4 people that are colleagues for the science initiative of the Million Mito Project. Dr. Vilar running the Genographic project for the last several years. And while the public participation phase ended, the scientific research for the people who opted in to share their science did not, and it lives on. And that is the information that we've combined with academic research that's been uploaded, the 1,000 Genomes Project and the full mitochondrial sequences at Family Tree DNA that will be informing the new tree. That's the goal. And I also need to say something here. I took this to Bennett Greenspan a little over a year ago, and I asked him for his support, because we needed support to sequence it. We needed permission for Paul's time, because he's an employee in Joran. And he said, go, just go, do it. It's important to science. And I'm really grateful. He's not listed here, but I'm really super grateful to Bennett for this opportunity, because we couldn't do it otherwise. So why does this all matter? Well, genealogy and genetic genealogy, they're all tied together. And while I know it seems simple enough, to quote Paul, I think it's not as simple as that behind the scenes, although we try and make it simple and lovable so that everybody can use it to find and love their ancestors. So the tree of mankind and womankind is two separate stories that we can trace for all of us. Well, men can trace them both. Women can only trace one, but then we find our proxy fathers and uncles and brothers to test. But it's something that we can trace our history beyond genealogy and back across the world from Africa through time. And we can figure out what those ancestors were actually doing at that point in history. It's an amazing story of success of our ancestors, because if any one of them had died, we wouldn't be here as the person that we are today. So for the Y-DNA, and I know it's like, this is mitochondrial, but bear with me for a minute. The Y-DNA tree growth was exponential. It started in 2010 with the YCC tree that had 441 branches. And then in July of 2012, Family Tree DNA, the Genographic project, started out with 850 branches, but they were going to test 13,000 SNPs. And I remember the day I got that call. I was actually on vacation on an archaeological digging for bones, of all things. I was there and I got this call from Bennett, and I'm like, what? I was sure I didn't hear. I'm like, I'm sorry, I've got a really bad, could you say 13,000? What? I mean, it was like so bleeding edge. We pushed that envelope so hard. So in November of 2013, we had tested enough people. We had pushed that to 6,200 branches using 50,000 testers results. That was amazing. I was so geeked out. I'm telling you why. I thought I died and gone to geeky genealogist heaven. And by February 2020, when we announced the Million Mito Project, the Y tree had over 26,000 individual tree branches. And that's because so many people have taken the big Y test at Family Tree DNA, and they started building out the branches. And we all know that story because we're genealogists. And now there's 500,000 identified and waiting to be placed in the right places as more people test. So that is a huge success story. So it went from a little tree to a big tree with citizen science testing and focus. And by that, I mean the testers test, the scientists figure it out, and the citizen scientists run the projects and encourage people to test and help them through the process. So that's what happened with the Y-DNA tree. And that's a success that we want to mimic with the mitochondrial DNA tree. Now, one thing about Y-DNA is men tend to be inherently super interested because it tells them about their surname line and they identify with that. Well, the surnames change in every generation with mitochondrial, so it's more difficult genealogically and it's sometimes less interesting to people, but it's not to me, obviously, or Miguel or Paul or Joran. So where were we with mitochondrial DNA? Well, in 2012, we had Build 14, which had 3,500 haplogroups from just over 8,000 sequences. Build 17, which is the current build in use, had over 5,000 haplogroups with 25,000 sequences. And in 2020, we realized we have over 500,000 full mitochondrial DNA sequences between Family Tree DNA, Genographic, NCBI, 1,000 genomes, or enough of the sequence that we could use it to at least be interested and to know we need to sequence it further. So in February 2020, we kicked off the Million Mito Project at Roots Tech. Unfortunately, my session and Paul's session took place at the same time, so we didn't get to see each other's sessions, but that's where we introduced it. And drum roll, please, you are the first people to know that we have submitted our first paper yesterday. So everybody just clap because this is a huge deal. Yeah, everybody's like, it's a really super huge deal. It's not the full tree, and we can't really talk about it yet, what's there, but this is with what we have, and we're still going for 1 million. We still want more people to test, because if we can do this much with what we have, just think what we could do if we could get 1 million or more people to test, which is why we named it Million Mito. So benefits of science and genealogy. It gives us all these tests give us something to work with genealogy. And the more people that test, the more we can refine these haplogroups. And the more refined the haplogroups are, the more closely in time we can see how these people match us without having to ask them for their mutations. Because if we rename all those little subclades with a leaf, then we know how they match us. We know how close in time they are, and we can look at where their cluster is. So it gives us more information without having to ask them for their mutations. We have the critical mass in this industry. There's over 30 million autosomal testers between the different, and I think it's significantly more, but we don't really know how many people tested at the same places. So there's more than 30. Just think if we could have even a fraction of those people test their mitochondrial DNA, what it would tell us, and how many brick walls we could break down and push back into time that we can't get any other way. We have success stories even with limited testing. So we need more testers, and we need more trees, and we need you. We need everybody here, and we need everybody to find their relatives and to test the relatives, so that we can feed that database and upload their trees and to list the earliest known ancestors, so that information is there for people to work with. So here's the participation. You can test if you haven't. If you previously tested at the HBR1 or HBR2 levels, you can upgrade to full sequence, because that's really what we need. The full sequence results will be used to expand the tree. We're adding them to the tree, will be reflected in the free tree that I mentioned earlier. That the free Y tree is there too, and that has the country flags of everybody with that haplogroup, not just the people that match you. That's a super underutilized resource. Genealogists don't even realize it's there a lot of times. We are going to define more haplogroups, and we are rewriting the tree of womankind. And I do want to say one more thing, and I didn't put it in here, but I am going to mention this. We can't just tack leaves onto the tree, because the problem is that, as Paul showed, the same mutation happens at various places in the tree. So let's say that I have people that I match, that I have differences in 3 locations, but some of those are the same, but not all of them. So which came first, this one or that one? And the further up the tree you go, the more mutations you find. So then you've chicken and egg, and we need to restructure and review the backbone and all the branches of the tree so that the leaves are really where they belong. So when we did build 17, for example, haplogroup A4 disappeared entirely. It's been incorporated under another haplogroup. But that's why we don't just tack leaves onto the bottom of the tree. You have to evaluate everything phylogenetically, because we now have so much more information than we had. So those branches are going to shift. And if you tack a leaf on today, just by renaming that leaf and tack it on the branch where you think it goes, you may discover, after evaluating everything, that's not where it goes. That's not the name even the branch should have. So that's why we can't just tack leaves on. And that's why it's not as easy as it seems like it should be, just looking at it superficially, which I did before I knew what went on behind the scenes. So it seems simple enough, but it's really not quite as simple as it seems behind the scenes. And we're working really hard to bring this to fruition. And the next question I know some of you are probably going to ask is, do the new owners, are they supportive? And they are. So they are supportive of this. They are behind this, that bought or merged, the family tree DNA merged, my DNA. And we have spoken with the CEO, the new CEO, Leo Rushberger, and he is very much supportive of this. So I just want to say that and thank them as well, even though they're not on the call today. But I don't think you have to worry about this not being completed, because they have picked up that and run with it as well. So I think that is it for me today. We're going to have more discoveries coming. We have set up a Mitochondrial DNA for Genealogy Facebook group where you can find Paul and Joran and Miguel and me answering questions and generally causing trouble. I love the Mitochondrial DNA Misfit, because we are kind of like the geeky people over there. And you're welcome to join us there if you would like to. If you have questions, Paul, do you or Miguel or Joran, if he's on, I don't see him, but I can't see everybody. Do you have comments, anything to say here?
That was better than I think anyone could have said. I would just echo what you already said, which is this is a people project. It's all of you guys. It's a populist project of you guys contributing your DNA to building the largest mitochondrial tree that's ever existed. And the paper we just released, I've been itching to say something about it. I know I can't, but it's a lineage that's so old, so late in the game, that it's mind-blowing that we got to where we are today. So just think, this is the beginning of the Million Mito Project. Think of where this is going to go if everyone contributes. So I can't wait.
Hopefully we'll be able to put it on a preprint server really soon, but we have to get through this initial little knothole first.